remarkable platform that brings together experts, researchers, and students from diverse domains to delve into the fascinating and complex world of microbial biofilm, tiny communities with huge implication across food safety, medical sciences, and industrial microbiology. We are privileged to have distinguished faculties and participants joining us for this enlightening journey of learning and collaboration. I request the dignitaries to please take the seats on the stage. Following this, I request you all to remain standing for the rendition of our Kulgeet as we honor our institute through this beautiful tradition.
विद्याधनं सर्वधनं प्रधानम् न चोरहार्यं न च राजहार्यं न भ्रातृभाज्यं न च भारिकारि वैकृते वर्धते नित्यम् विद्याधनं सर्वधनं प्रधानम् शिक्षा दीक्षा की परंपरा से राष्ट्र का हुआ सदा उत्थान। कौशल और नव सृजन से सजा हुआ गौरव अभियान,
सजा हुआ गौरव अभियान।
ज्ञान विज्ञान की शक्ति से हम नवमंडल में हुए स्थापित। सीखा है इतिहास से हमने नव तकनीकों में हुए समाहित, नव तकनीकों में हुए समाहित। सगम सानि पम गमरि सनि सनि संस्कारों की शक्ति को जब लिया है युवा ने पहचान। हम सब बने राष्ट्र के रक्षक, आओ करे नव भारत निर्माण, आओ करे भारत निर्माण।
कला योग विज्ञान विधि का विश्व विश्वविद्यालय देता ज्ञान, तन मन और जीवन सुचिता का देता है यह शुभ वरदान।
जन्म लेकर जिस धरा पर शौर्य करता शीश बलिदान,
अपनी मेहनत से माटी को सोना कर दे जहाँ किसान।
हर की धरा हरियाणा पर स्थापित अपना कुल महान। शिक्षा और स्वावलंबन आओ ऐसे कुल को करे प्रणाम, ऐसे कुल को करे प्रणाम, ऐसे कुल को करे प्रणाम, ऐसे कुल को करे प्रणाम।
To formally begin the day's proceedings, I now invite the Dean of CIAS, Professor Dinesh Kumar Gupta sir, to deliver the welcome speech. Professor Gupta is a senior professor of our university and is a faculty member of Department of Library Science and Information Science, and presently the Dean of our school and chairman of this program.
Thank you so much. Good morning, everyone. Honorable Vice Chancellor, Professor Tankeshwar Kumarji, the guest from US, Professor Bhunia ji, another invited speaker, Professor Dr. Shukla, the Professor Sunita Srivastav ji, Dr. Gunjan Goyal, the coordinator of the program, head of the department, Dr. Beniwal ji, Director Research, Professor Neelam Sangwan ji, all the heads of the departments of different part of the school, researchers, faculty members and students. It's really a great pleasure for me to welcome you all in this very important program organized under the series of GIAN programs. And this department of microbiology, has been front runners in so far as conduct of GIAN programs is concerned. And Dr. Gunjan Goyal is also coordinator for the university to have these programs in whole of the university. Perhaps this is the first program which will go for 2 weeks. Otherwise, GIAN program takes 1 week's time. So this is a new dimension for the GIAN program for the university. So I take a privilege to welcome our Vice Chancellor, Professor Tankeshwar Kumarji, who has always been kind enough to grace such occasions. We welcome you, sir. I also take an opportunity to welcome Professor Bhunia from very prestigious University of US, Purdue University, who has come all along with taking so long journey, and he will be with us to the and of course for the interdisciplinary researches, because this school is having interdisciplinary kind of subjects like microbiology, biochemistry, biotechnology, food and nutrition biology, then environmental sciences, just for the sake of the students or researchers who are coming from other universities. So I welcome you all again for this program. Thank you so much.
To provide an insightful overview of this program and its significance, I request the course coordinator, Professor Gunjan Goyal, to take the stage.
Thank you, Anusha. As we all know that GIAN, the word in Hindi and Sanskrit, it means knowledge. So, and it does, the knowledge doesn't have any boundary. It can be through, like nowadays, through the mass communication, we can spread knowledge through different parts of the world. So this initiative, that is Global Initiative Alliance Network program, it is a flagship program of Ministry of Education, where we host some faculty members who has from the different institutes abroad, and they conduct the course for 1 week or the 2 weeks, that is provision from the GIAN only. So the main aim is to augment the country's existing academic resources, accelerate the pace of quality of research, reforms, and elevates our country's academic and scientific knowledge. At CUH, we have already completed 16 GIAN courses, which were under phase 1, 2, and 3. And in the last phase, this fourth phase, we have been awarded 4 GIAN courses at our university, and this is the second program which we are conducting right now. And till date, we have total received 20 GIAN approved courses from the different departments of the university, from the management studies, biotechnology, biochemistry, nutrition biology, environmental sciences, social sciences, and even education. This GIAN course on microbial films, it is having another importance, because in microbiology, the microbial films is posing a great threat to the mankind. Maybe it is because of the food infection, foodborne pathogens, food spoilage, and many other things in the environment also. So we have different applications of this applications, or you can say that we have positive and negative sides of microbial biofilms in microbiology. So that's why we have designed this course with Professor Arun Bhunia. And in this course, we have 56 registered participants from different universities and colleges across the country. And some of few are 31 participants are from universities of colleges from University of Burdwan, Qazi Nazrul University, Bhardak Autonomous College, Odisha, Chandigarh University, Manipal University, Bundelkhand University, Shri Vishwakarma Skill University, Chaudhry Bansilal University, Wilfred College, Jaipur, and 25 participants are from our own university. Under this program, we have eminent speakers, Professor Arun Kumar Bhunia from Purdue University, who is the course instructor for this course. Along with him, we have Dr. Sudhir Kumar Shukla from BARC, Dr. Deepshikha Chakrabarti from ISC Bangalore, Dr. Krishnan from RCB Faridabad, Dr. Kiran Kanti from NABI Mohali, Professor Antaresh Kumar, Dr. Pooja Yadav, Professor Vikas Beniwal, and Dr. Jitendra Kumar Sen from our institute. And to accomplish this course, we have scheduled 27 lectures and 10 practical demonstrations in this 2-week program. I hope that deep insights from the experts of this area in the emphasizing the role of biofilms in the food and health will be delivered during this 10-day course, and the participants will get benefit from this, and they will earn 2 credits of complete 2 credits during this 2-week course. Thank you.
As a token of our deep appreciation, we will now felicitate our esteemed dignitaries. I request Honorable VC Professor Dr. Tankeshwar Kumar Sir to felicitate our speaker, Professor Arun K. Bhunia, the foreign faculty from Purdue University, who is also the course coordinator for this program. Now I request VC Sir to felicitate Dr. Sudhir Kumar Shukla from BARC. Now I request our Dean CRs, Professor Dinesh Kumar Gupta Sir, to felicitate our Honorable VC Sir. I now request Professor Vikas Beniwal to felicitate our Dean CRs, Professor Dinesh Kumar Gupta, sir. I now request Professor Vikas Beniwal. Now I invite Head of the Department of Microbiology, Professor Vikas Beniwal, sir, to introduce our Honorable Vice Chancellor, Professor Dr. Tankeshwar Kumar, sir.
A very good morning to all of you. It's my honor to introduce our Honorable Vice Chancellor, Professor Tankeshwar Kumarji. He's joined Central University of Haryana in July 2020. So he's on the verge of 4-year completion as a Vice Chancellor of this university. And we are very blessed to have him as a leader, as a mentor, as a guide. Before joining Central University of Haryana, he served 2 tenure in Guru Jambeshwar University of Science and Technology as a Vice Chancellor. He also served as an additional charge of Vice Chancellor of Deenbandhu Choturam University of Science and Technology, Murthal, Chaudhry Bansilal University, Bhiwani. He has more than 35 years of experience, which includes teaching, research, and educational administration, and contributed more than 200 research papers in national and international reputed journals. He has 3,000 international citation with an H index of 27 and I-10 of 58. His research area is theoretical and computational condensed matter physics and nanotechnology. He is the first UGC professor in India in FRP. He has guided more than 13 students for PhD degree. He is a fellow of International Academy of Physics, Science, Physical Science, and member of National Academy of Science, Allahabad. He is a member of General Council of National Board of Accreditation, Chancellor nominee of Selection Committee of Appointment to Teacher Post of CRSU Jeend, Chancellor nominee on the Selection Committee for the Appointment of Teaching Post in Kurukshetra University, Member Academic Planning Board, Kurukshetra University, Member Planning Board, SVSU, Palwal, Member Planning Board, Guru Jambeshwar University of Science and Technology, Member Academic Planning Board, Gurugram University, Member Academic Planning Board, MDU Rohtak, University Court, Member CBLU, Bhiwani, Executive Council CBLU, Bhiwani, Executive Council SVSU, Gurugram Finance Committee, Central University of, Finance Committee, Central University of Punjab, and Executive Council JC Bose University of Science and Technology, Faridabad. He has been a member of Senate and Syndicate of Punjab University. He has been a member of various committees at state and national level like UGC Examination Reform Committee, Chairman Committee to establish First Skill University of Haryana, although it's a first skill university of country, Planning Board YMCA University of Science and Technology, Faridabad, AC of JC Bose University of Science and Technology, Member Board of Governors of IIIT Sonipat, Chairman of Committee of Student Election in Haryana, Chairman Member of NAC Peer Team from time to time, and Search Committee for Selection of Vice Chancellor and various universities. He has been awarded associateship of ICTP Italy from 1997 to 2004. He has been research leader ICTP Italy from July 1993 to August 1993. He has visited abroad in a number of countries for you can say more than 17 times in Europe, Africa, and America, in various countries in these continents. He has been awarded with the Ambassador of Green Planet Award 2022, Shiksha Ratan Award 2022, Vice Chancellor of the Year 2022, Elite Academician Award 2022, Golden AIM Most Dedicated Vice Chancellor Award 2020, Vidya Sagar Award 2017, Lifetime Achievement Award 2016, Appreciation Award by Federal of Corrugated Box Manufacturer 2016, Education Leadership Award 2016, IBISH Award of Amity University. Under his dynamic leadership, Central University of Haryana has been included in the expert heritage body by Ministry of Council, Government of India for forming a heritage bylaw of Central Protection Monument and Protection Area. He has been associated with various social organizations also like Bharat Vikas Parishad, Shiksha Mandal, Vigyan Bharati, Seva Bharati, Savdesh Jagran Manch and many more. With this, I request Vice Chancellor sir.
Thank you very much, Professor Vikas. First of all, I thank Professor Arun K. Bunia from Purdue University for accepting the invitation for GIAN program to be conducted at Central University of Haryana. And I know this program was started in around 2016 by the Ministry of HRD, that time it was called HRD, to have foreign faculty in the university where we can have some particular topic to be discussed thoroughly for at least 7 days by the foreign faculty and train over young students, researchers and teachers on a particular topic. And I believe that the participants, those who are attending this program, will take it seriously to meet the expectation of the ministry. That means we are to really learn the topic and utilize it for further progress of your research career and for the, I will say, for the country. And the topic which I look at here is microbial biofilms in food processing, health and disease. If I look at the topic, at least 3 things are very, very common to everyone, common citizen, because everybody is using the processed food. Almost everybody nowadays, right? Whether he belongs to some village area, even then he is using somewhere. Health and disease, obviously. So something is very important, common to everyone. So we should take this topic seriously. You will be, this microbial biofilms, that may be the, all of you may be knowing, but not everyone. So you have to really understand, the really new techniques, how the food processing, actually is done, and how it could be, make many people say that हमें food process जो है process किया हुआ food नहीं खाना चाहिए. हम food processing की बात करते हैं एक तरफ कि किसानों का जो हमारा, यदि हम उनका सामान खरीद कर उसको प्रोसेस करके बेचेंगे तो किसानों की इनकम बढ़ेगी. दोनों तरफ हम बात करते हैं, दोनों ही पहलू हैं हर चीज़ के. तो उस पहलू को बीच में जो काम करते हैं वो रिसर्चर्स हैं. They are the researchers. Now they have to see that such things become viable, become sustainable, become useful for the, I will say, everyone, right? So that is where I see the importance of research is, and especially the bioscientists are being playing wonderful role, especially we call it sometimes we say that we take the food for for over hunger, food for our to live in, but sometimes we take the food we have to calculate the medicinal value of every food. Then you start taking. So you start marrying. An age will come when you start taking the food as a medicine, and also you taking the medicine with the foods. Right? That is the the part of the things will become when you taking the medicine with the food, and you will taking the food as a medicine also. Right? So you have to really work on such things, so that we take the I will say food as very, very important. What is required in the for a human being is essentially the two things: food and sleep. If you have a food and sleep, that's the end of the thing. Rest of everything is there, right? So that the food is what kind of food you take. So that is where you look at or how you sleep, where you sleep. That where you look at your lifestyles matters. If you look at the lifestyles are different. So that is the which kind of food, what kind of food you are taking, and where you are sleeping, why you with what comfort you are sleeping. That is what we look at the lifestyles, right? So it's very essential that we carry out meaningful research, so that our food habits, whether it is whatever it is, right? So that should be should not harm our health, or should not harm or create the disease, right? So sometimes people say that we are using this kind of food, processed foods, we have, so we got disease we have disease is because of this processed food, right? So we have to really think of that, how we can contribute towards the sustainable I will say the research, where you can have over health, and we can have the cure to the disease. But it's very, very difficult to find out in allopathy to whether you can cure some disease or not, right? But anyway, so I am really thankful to having such a program in the Central University of Haryana. I understand that we have the participants from other parts of the country. We have some participants from the university, so I hope that they will have a comfortable stay here. And I am thankful to our Dr. Sudhir Kumar Shukla. He is from Bhava Atomic Research Center, and we have been associated with the Bhava Atomic Research Center from our, I will say, childhood of the research, right? So if we look at starting from maybe in 1986 or 7, we have been going to the Bhava Atomic Research Center. But that time Bhava Atomic Research Center was carrying out research in I will say physics, different branches of physics. Obviously the most important was the nuclear physics, and then atomic solid state comes up, and then some chemical lasers and all these things were coming up. But nowadays I think that we have all come all sciences are coming closer. We are all maybe working on the biological sides of the aspects, and the nutrition biology also. We know even the effect of the radiation on the food items, and how it's improve its shelf life, or it's improve the quality, or improve the things. So all such kind of research are coming up. So we are having a time where we say that all the research areas are combining to to the extent that how we can help the mankind. So with these words, I really thankful to Dr. Sudhir, and I know there are others, those who will be joining later on. So I am thankful to all of them, those who are joining later on in this event. And this program is being the coordinator of this program is Professor Gunjan Goel, and the convener is Professor Vikas Beniwal. I am thankful to him for bringing Professor Arun K. Bhunia to this Central University of Haryana to interact with our young students or the faculty members of India. Thank you very much.
Thank you, sir. It is now time to introduce our speakers. The course coordinator whose presence greatly enrich this event. Professor Arun K. Bhunia received his Bachelor of Veterinary Medicine degree from India, PhD from University of Wyoming, USA, and postdoctoral training from University of Arkansas, USA. Currently, he is a professor of food microbiology at Purdue University and an adjunct faculty member of Department of Comparative Pathobiology, Purdue Institute of Inflammation, Immunology and Infectious Diseases and Microbiology, and Infectious Disease Training Group of Faculty Professor Arun K. Bhunia, PhD, Purdue University, Interdisciplinary Life Sciences Program. His expertise is in the area of microbial pathogenesis, probiotic bioengineering, and food-borne pathogen detection. To date, he has co-authored 228 research publications, 2 test books, edited 4 books, and delivered over 178 talks in national and international venues. He is awarded 7 patents. He is serving as the editor-in-chief of Foods and associate editor of Frontiers in Microbiology, PLOS ONE, Frontiers in Sustainable Food System, and BMC Microbiology. He served in many of the national and international committees on food safety and has been awarded with various prestigious fellowships and awards. I request Professor Arun K. Bhunia to kindly share few words of guidance and encouragement with us.
Thank you very much. Oh, sorry. I guess I'm under jet lag, so I'll be a little bit slow, but I'll try to communicate to you because of the travel, obviously. So, first of all, I'd like to thank Professor Kumar, Vice Chancellor, for having me in your campus, and also Gunjan. And he contacted me, of course, for many years. We've been in touch, and asked me whether I would want to participate in this course, and so I'm delighted to be part of this this course. So before coming to Haryana, Mahendragarh, and I asked him how is the weather. So he told me it's going to be 40-45 degree, and then I start the weather or the temperature at Purdue, which is West Lafayette, very close to Chicago, and we are having temperature like a 15 or 20 degrees C. Then when you say 40, 45, I need to have 20 degree of adjustment, right, to be here. And so naturally I felt that it's pretty hot. But then I started thinking, last year I was traveling to a place, I don't know whether you heard about this place called Death Valley. Any of you heard about Death Valley? That's in, probably you heard about also, your son is there nearby. In San Francisco, so it's in California, Nevada border. And last year summertime I was traveling, which is very daring, and the temperature we recorded in my car is showing 52 degree, 52.2 degree Celsius. So, and you couldn't stay barely 5 minutes outside the car, outside the air conditioning. So I was thinking, wow, that's very hot. The highest temperature, I think they recorded 55, I think. That was the temperature. And if you look around, it's like a snow. That means this is the salt, all water, turned into salt. And so it's pretty beautiful, but you don't want to be outside. There are some people died because they were there, getting out of their car, started walking or venturing out. There are, I think, 2 people died before we went to that place. So it reminded me, it's not quite that temperature here, but it's pretty close, right? And so it reminds me the climate change, right? And that's the temperature, the global warming. And it's pretty obvious when I visit India, looks like pretty high temperature. So then I started thinking, being a microbiologist, what is the relationship between climate change and microorganisms? Think about it. Microbes like to grow, right? Higher the temperature, their growth rate goes high. And then think about this. The microbes, their community in all those different ecosystems, whether in the environment or in the food or in the patient, microbes all causing varieties of diseases and also varieties of applications. And so the biofilm being part of the source of these organisms. So more temperature rise, climate change, and then you'll find also the microbes find a way to adjust to this high temperature, and one way they like to huddle together so that they don't die each other, they become a community. And that's the biofilm, right? And that's what we're going to talk for next 2 weeks of lecture. We'll talk about varieties of topics related to biofilm. It's not one single entity, so we have different expertise also in the team to teach you about the biofilm. And the temperature rise also directly links to foodborne outbreaks. There are a lot of data available right now. As the global temperature rise, every year we are finding more and more foodborne cases. Especially there are cases like Campylobacter, one of those organisms caused diarrheal diseases. And there are clear record that the climate change and foodborne pathogens, even not the foodborne pathogens, also even the toxins, the algal bloom in the water, and a lot of these toxin conditions begin to increase. So hopefully we'll be able to communicate that to you during this next 2 weeks, and I think we're going to have a lot of fun. We'll talk about biofilm and microorganisms, and as introduced, that we have written a couple of books, textbooks for the students, and this is one of the book which is called Fundamental Food Microbiology, and we just published this year in January, and this is called 6th edition. And my major professor, Professor Vivek Ray, he actually started this book, and then I took over and started publishing it. And this is the 6th edition. It's published by CRC Press. And so I thought a lot of those contents we will be talking probably from this, some of this coming from this book, and also to get general information about foodborne pathogens, spoilage organisms, beneficial microbes, the microbial world in the food systems. That's pretty fascinating. So I brought this book as a way to communicate to you that we'll be talking about this, and also to extend my appreciation and recognition that Professor Gunjan Goyal asked me to come here. I thought most appropriately to offer him come here. This is for you to keep it and to share with the students. Okay.
I have been studying this book in my master's also.
But that was the previous
That was the first,
edition.
yes.
So this is the 6th edition. So hopefully it will be useful for you and your students. I'd like to thank you, and I'm sure I'm going to see you very soon with the introductory lecture on biofilm. Okay. Thank you. Thank you very much.
Thank you, sir. Our second guest, Dr. Sudhir K. Shukla, is a scientific officer at the Biofouling and Biofilm Processes section, Baba Atomic Research Center, specializing in microbial biofilm research. He holds a PhD in life sciences from Homi Bhabha National Institute, Mumbai, India, and also serves as an assistant professor at Homi Bhabha National Institute, Mumbai. Dr. Shukla's research focuses on the formation and control of biofilms, particularly those formed by Staphylococcus aureus. His work has significantly advanced understanding of biofilm associated proteins and the development of strategies to disrupt biofilms. He has authored over 34 journals publications, contributed 9 book chapters, and has an H index of 19 with approximately 1500 citations. I request Dr. Sudhir Kumar Shukla to kindly share a few words of guidance.
Good morning, everybody. It is truly an honor and privilege for me to share the dais with such esteemed Professor Dr. Arun Bunia and other guests. First of all, I want to thank Vice Chancellor Dr. Tankeshwar Kumar. I just had a chat in morning that he was previously associated with the BRC. It was such nice thing to hear about, and really I want to thank Professor Gunjal. He was behind me that you please attend, otherwise I would have missed this one. He made all the arrangement to pick up me from airport and all the way come here, and I really wish I could spend more time to be here and learn many things from Professor Bunia and other dignitaries. But I'm sure that you will learn a lot about microbial biofilms, especially UG and PG students. I would say that take microbiology very seriously, not only for your course but in life as well. Many things you will read in microbiology, but about biofilms you will read very less things in about biofilms. You won't find much, because recently I have encountered many students who are really like microbiology, but still they don't know much about biofilms. That I have seen. So to learn about biofilms, this is the one incident where you can really learn many things about biofilms. And you have been seeing that many conferences happens across India and international, but mostly they are in general for the microbiology. Last year only two, one is from us. We have formed a biofilm society. Our society is Biofilm Research and Microbiome. People who are working in that area or interested in that area can join. Another one is India Biofilm Society. So if you are really interested in that biofilm work, you can join any of this society. And I would say that take opportunity to learn as much as possible from this coursework which is being arranged from Professor Gunjan Goyal, because this kind of opportunity is really rare, where you focusly learn about biofilms and hands-on experiments you will be able to carry out, and it is going to be really helpful in future, that I would say. With that, once again thank you all for being here. I hope that we'll have wonderful learning together. Yeah, thank you very much.
Thank you so much, sir. I now request our Dean Research, Professor Neenam Sangwan, to please come on the stage and share some wisdom.
Very good morning, all of you. Honorable Vice Chancellor, sir, esteemed speakers from abroad, Professor Arun Bhunia and Dr. Sudhir Shukla from BARC, Bombay, our Dean Sias, Professor Dinesh Gupta, and the team from Department of Microbiology, Professor Gunjan Goyal and Professor Vikas Beniwal, Dr. Pooja Yadav. I'm very happy to be here and attending this inaugural session on GIAN program. This Department of Microbiology has already, taking lead in all the directions of teaching and sharing, and especially in GIAN program, as Professor Gunjan Goyal has told us, there are so many GIAN program, and the idea behind it is that the knowledge from, as our Honorable Vice Chancellor sir also shared, that the knowledge from the people who are accomplished, who are coming from top labs, they are shared with our younger generation. So they are equipped for and ready for the future. So I am very, very happy and thankful on my behalf to our Honorable Vice Chancellor sir, who does not leave even single opportunity for promoting, for encouraging our staff and our student, young student, for research opportunities. So he is always with us. You can see, to motivate all of us, he is always here. And I am very thankful to our external speakers who have come all the way in such a hot weather, as you have seen the example given by Professor Bhunia, and that how the climate change is changing everything. So from when he was speaking, I was also inferring that this is a time, this is a GIAN time, we are going to learn how a small, tiny, single cell, small structure is able to combat everything which human and which multicellular eukaryotic systems are feeling, and how they are adapting. So that this is the era, this is the time when we need to be flexible, we need to be adaptive, and climate change, this is happening, it is inevitable, it is happening every day. things are changing, temperature is rising, and so many other climatic conditions that we all are watching. But there is a lesson to learn that how a single cell small bacteria, which none of us can see, when they are in thousands of number in a cell, then we are able to see that too during by the microscope. So there is a lot to learn. There is a lot to learn from our speakers who have come, and many more speakers who are likely to join. So with these words, I congratulate the department for this good work, for this exceptional work, for having this GIAN program for all of us. I thank our Honorable Vice Chancellor for giving this opportunity to all of us. Thank you very much.
Thank you, ma'am. As we come to the conclusion of this wonderful inaugural session, it is time to express our sincere gratitude to everyone who contributed to making this program a reality. To deliver the vote of thanks, I invite Dr. Pooja Yadav to take the stage.
Thanks, Anita. I, Dr. Pooja Yadav, on behalf of the faculty members of the Central University of Haryana, extend my heartfelt thanks to Honorable Vice Chancellor Sir, Professor Tankeshwar Kumar, for his gracious presence at the inauguration of a GIAN course titled Microbial Biofilms in Food Processing, Health and Diseases, a 10-day program scheduled from May 26 to June 6, 2025. Sir has been very kind in supporting such kind of academic activities in the university. Your unwavering support for such academic initiatives and for facilitating this course is deeply appreciated, sir. We are truly grateful for your encouragement in making this activity possible. I am also thankful to Professor Arun K. Bhunia, Purdue University, USA, and Dr. Suhir Kumar Shukla from BARC to be the resource person in today's inaugural session. I am thankful to Dean Research, Professor Neelam Sangwan, for her unwavering support, and to Dean Sias, Professor Dinesh Gupta, who serves as the course coordinator and has been a great source of motivation throughout. I am thankful to Department of Microbiology for organizing this activity. I extend my thanks to all Dean of Schools, Head of the Department and faculty members, Professor Sunita ma'am, Professor Antaresh, Professor Pavan, Dr. Tejpal, all of you for your valuable time to join us today. A special note of appreciation goes to our enthusiastic audience, our students, for their active participation and engagement. I would like to acknowledge the exceptional efforts of the students, staff members and the ICT team whose dedication has ensured the smooth execution of this program. I would also like to thank Professor Baniwal, the coordinator of this course, for his invaluable support and guidance. To our distinguished guest, I once again extend a warm thank you for your presence and support. Now I kindly request everyone to please rise for the national anthem.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
I request you all to please come forward for a group photograph.
किसका? हाँ हाँ नाम चेक कर लिया मैंने। हाँ हाँ प्रोफेसर टंकेश्वर। अरे प्रोजेक्टर बंद करना है एक बार ना। नहीं नहीं एक बार। रोहन निखिल नीचे, करुण नीचे, सुपर्ण
Tea is being served out.
Okay. Well, let's get started. So, well, are you still in the morning, right? So I should say good morning, right? Are you comfortable? Are you, or is it hot? Too hot? Well, even if it is too hot, I can't help you. I don't control the air condition, so um, I like to move around. I don't know whether you'll be able to. Do you have another mic? That, well, that'll be good though. Do you have one? Oh no, you don't have the one. Yeah, this one. Oh. You control the volume. Okay, good. Can you hear me? Can you hear me? Okay. Okay. Can I think you can hear me, right? No? Yes? Okay. Sorry about that. I know we're going to, I'll be talking to you for next 2 weeks, right? So this is the beginning. Make sure we got the right beginning. So how many of you are undergraduate students? I think, okay, raise your hand. So those who are undergraduate, how many microbiology courses you had? You all had microbiology, right? Except this was, you didn't have microbiology? Okay. And I was assuming rest of you are postgraduate, right? Okay. You didn't say anything, but so when I teach usually, well, this distance is too far. Are you going to meet all the time in this room or different room? This room? Oh, okay, so they are too far away from me. So I have to speak loud, right? So what I want, if you have any questions, any point, feel free to raise your hand or just yell or scream, okay? So we want to make it interactive in a sense that makes sure all those things I'm going to talk, you follow, right? Otherwise, if I keep on talking and you don't have any clue what I'm talking, that's not good. It will be just, I will be spending a lot of energy for nothing, right? But I want you to learn something, right? With this course, even though it's going to be kind of like an intensive, right? I mean, usually we take like a 1 semester or 2 semester long to learn all of these materials, but you're going to learn very quickly, right? In a very short period of time. So you need to figure out how to compartmentalize your information, right? And there will be a lot of information, so then you have to figure out what is the most important message in that topic I will talk, right? So the way we will start presenting the materials is that there will be a time I'm going to go like a level, a little bit superficial, means up there, right? Based on your understanding, I'm going to go deep and deep and deep and deeper, right? And so I will go up to a certain level. If you feel uncomfortable, of course, unfortunately, you have to take that challenge, right? And so you got to go and read. And so these slides would be shared with you, right? You are going to get these slides. So you can go over the slides. And when I come back next time, definitely I can take time to address your questions. Will you follow me? Okay, so again, the whole idea being is that this is a special course designed for you to learn about biofilm, right? And the goal would be that make sure you understand, right? Okay, so that we are clear on that. And other aspect I want to mention is that these lectures, I have prepared about several lectures. You already have the syllabus, right? Correct? So there are many speakers besides me will be talking to you, and there are information going to be redundant or overlap. But I think that's very good, because in the short period, you don't want to get all the information from me, but there are many expertise, those who are working on specific areas of this biofilm project, and you will gather a lot more detailed information. So don't feel bad that if you heard from me, then other speaker talking about it. Okay? So just think that this is going to reinforce your learning from a different viewpoint, different perspective. Got it? So that's the beauty of this course. I think the way the course has been designed by Professor Goel, and I think I like that, because I'm not the expert. I don't know everything. Okay, I'll just tell you in front, up front. So based on that, we'll get started. So though there are two chapters we are going to talk, introduction and significance. So what I did, that I combined them together. Okay, so whatever I can finish today, I will do that within the time window, and I try not to go over the time, right? And then I will talk about significance as we come along. And like, there will be a lot of repeats also. If I talk some information today, then you will see that I may be talking about that again another 2-3 days later. Okay, so those are the background information. And of course, you are going to have also exam, right? So that will be the last day. Okay, so prepare in a way so that you take the exam and you pass. Okay, so the way I teach my students, I talk to them the day one that I'll give you A on the day one. Are you follow me? So your grade is A. That means that's the highest grade, right? Because all of you are in the academic system. In our system, we call it 4.0. Okay, so I give you A to start with. If you give me a black paper, I'll give you A. The question is, can you maintain that A at the end of the class when you are taking the final? Are you getting it? So I give you A. So all of you have that A grade to start with. Now we'll see if you can maintain that at the end when you're taking the exam. Okay? So that's how I challenge you. So that means you try to keep it. And if you fall behind, that means, of course, that's what you got. All right. So let's talk about the introduction. First of all, I know you're going to listen to me for next 2 weeks, so it's good to know who am I, where I come from. So who am I? You already learned a little bit about the introduction. I don't want to spend any time, but where I come from, in terms of US, this is the map of US, and I'm sure you are familiar with that. And the state called Indiana, and the university is called Purdue University, and it's pretty located close to Chicago. This is Illinois, and it's the Great Lakes right here. So we're kind of like the middle of the state somewhere, but it's pretty north also compared to southern region. I actually, as a graduate student, I started at Wyoming. This is right here, and Wyoming is a wide open space. It's the mountainous area, and this is where I did my college, PhD. And from there, I actually moved to Alabama. Before that, I was in Arkansas, University of Arkansas. Then went to Alabama as a faculty member at Huntsville. That's where the space program. And then, of course, moved north in Indiana. So I've been there ever since for the last 27 years. So total, I've been spending US for 40 years. Okay, so I've been there for a while, but at Purdue University for 27 years. Anyway, so the campus is beautiful. Just to tell you a little bit, these are some of those pictures. This is a huge campus. We have a strong engineering program, agriculture, veterinary medicine, then business, arts and science and all kinds of programs. So it's a huge campus. These are some of those pictures of the campus, and our logo is called P, which is the Purdue. Purdue, we call it Boilermaker, and this is actually the train it represents. And just a little bit of history about Boilermaker. They used to make actually train engines, okay, and that's why they are called Boilermaker, okay, steam engine. And so we have a car actually representing that Boilermaker. There are some statues in the campus you will see all around. This individual, his name is John Purdue. He gave us the land, and that's how the university was named, called Purdue University. And anybody recognize this individual in the middle? Probably not, but you know he's the first man on the moon, right? Neil Armstrong. He graduated from Purdue University, and so Purdue is a home for many astronauts. They are also currently a very active program, and around the campus, you walk around, there will be a lot of statues. So you heard about the book, the Fundamental Food Microbiology. That's the textbook for undergraduate students, those who are learning about food microbiology, foodborne pathogens, spoilage organisms, beneficial organisms, and well described. And there are many undergraduate textbooks, but this is the 6th edition. And you know, Dr. Goel has a copy if you want during this time, if you want to see or borrow, you know, and you are welcome to do that. And then there's a graduate level course called Foodborne Microbial Pathogens. This is completely for graduate level students, means we call it postgraduate. And it's more upper level, describing mechanism of pathogenesis for pathogenic organisms. That's what is being covered in that book. Anyways, these are some of these resources for you to understand. So the way the course is designed, I'm not going to read everything, but you will see that these are the in your syllabus we are going to cover, right? And then including guest speakers, they are going to also cover many part of it. So to start with, what is biofilm, right? And how the biofilm structure look like, and then we are going to talk about also how they help persist organisms in different environments and how they contribute to the disease. Then we'll talk about the mechanistic aspects. Then we'll talk about how do you detect them, assay them, then how do they contribute to the antibiotic resistance. And these are some of those factors in pathogens, right? Then the biofilm also produced by non-pathogenic microbes, okay? Also the beneficial microbes. How do they use that biofilm strategy to help our health, okay? So there are a few things you have to remember. There are good, there are bad, okay, and there are some in between. So that means biofilm, don't take it as a negative way always, okay? Sometimes extremely beneficial, extremely helpful, okay? So some of those aspects will bring that up. And then how do you control or prevent biofilm formation? And then towards the end, we'll talk about the advanced detection methods for biofilm. How do you detect them? What are the current methods? What are the current trends of detecting these biofilms? And in the laboratory modules are designed for you to learn some of those techniques, okay? Other thing I want to tell you before I start is that there are some of the research. We've been doing a lot of research also on biofilm. I will not say a lot because our lab is designed for working with the pathogens. And so time to time, I'm going to introduce some of the research we have done and how that relates to the biofilm, or how biofilm formation helps the disease process, okay? Or biofilm formation can help improve your health, okay? So I'm going to introduce some of our research with the data, okay? So that way you get a more like a broader picture of biofilm, and at the laboratory level or research level, how do they actually work, you know, experimental level, how do they help, or how do they, you know, cause detrimental effects, okay? So this is how it is designed. All right. So let's talk about today's topic. We're going to talk about introduction and significance. So I'm going to talk about importance of biofilms in the food industry, okay? Also in the environment. Then we're going to talk about biofilms in food spoilage. We're going to talk about sustainability aspect. We're also going to talk about the disease outbreaks and then food safety concerns in biofilm forming zoonotic diseases. So these are some of the topics I'm going to cover today. Okay, any questions so far? All right, let's talk about how do we start, right? This is a picture of example of biofilm I just started using. Okay, this is called a park called Yellowstone. You heard about it? This happened to be in Wyoming. That's where I originally started, and it's a beautiful picture. This is a big park or national park. Okay, and if you go to this park, you can spend like 2-3 days. Still, you will not be able to completely visit the whole park. Okay, but for people who are working on biofilm, this is the Mecca, right? We call it Mecca, right? Mecca. People, those who are religious, they go to Mecca, right? The Islamic culture, right? And so for biofilm researcher, those who are especially environmental biofilm study, this is a perfect example. So this is one of the examples. You can take a look at all those formation of the color, beautiful color, and this water actually hot water. Okay, it's a geothermic activity, and you can see all those colorful organisms they are growing, and the way they sustain their life as actually forming the biofilm, right? And Dr. Shukla is going to talk about a little bit also environmental side of biofilm. So this is a classic example, the biofilm, right? Let's look at another one. So this is continuous geothermic activity, the water coming out of the ground, and it's like a volcanic activity, but it's the water, hot water keep coming, and you can see this formation, the structures actually contributed by microbes. These are all microorganisms, forming a nice tight structure, and then of course they mineralize and they calcify, and eventually this type of solid structure is hard to break. It's not fragile. It's extremely solid structure. So these are examples of how the environment or microbiology or biofilm look like. This is also Yellowstone, and this is also another one. You can see this. This is called bacterial river. Okay, if you look at the bacterial river, it's very colorful, right? And you can see this is formed by algae, the bacteria. They're all collectively growing together and forming a river. And so that's also a prime example of biofilm in nature. Okay, and this is again in Yellowstone. And if you go to that park, obviously you will be just amazed how this microbial activity from this natural structure is just breathtaking. It's beautiful. You can spend hours and hours watching these, and you'll be amazed that how nature take these microbes or microorganisms under these conditions, and they are growing and they are surviving, and then forming this beautiful biofilm-like structure. Okay, so this is an example. How about this? This is a pipe, right? Water supply, and you can see this built up. This is also part of the microbial activity. And they are also forming biofilm or biofilm-like structure. So, best form of definition of biofilm. One of the things I learned from my professors that microorganisms never exist in isolation. Like we as a single entity can be present, right? But microorganisms in nature, they never a single cell. They are always biofilm former. They are with a community with other microorganisms. So always microbes exist as biofilm. So that's not a surprise. We are learning it. If you look at the history of the biofilm, I mean, you can go back to Leeuwenhoek or Pasteur. They knew the microorganism exist on surface, they are actually forming certain biofilm. They may not have coined the terminology as a biofilm, but it exists for centuries. Okay, and in environmental microbiology, obviously, people started to understand that microorganisms present on the environment, they are in the form of biofilm. Okay, in the medical science, medical microbiology, clinical applications, the first, I think the report was 1985. They reported that the biofilm actually responsible for causing many diseases. And the first disease they studied, that is a lung disease called cystic fibrosis. They found Pseudomonas is the main organism responsible for that disease. So that's when the medical biofilm terminology was established in 1985. Okay, and ever since then, all those other field of areas, okay, environmental, starting with soil, and then you can say plant, then also in the food sector. I think I would say much early, late 90s and early 2000s, people started to study biofilm and their implication in diseases and also foodborne, contamination, and so food processing plants. All of those factors begin to be reported during that early 2000s. So the take-home message is that the biofilm is a natural process for microorganisms, and they form called polymicrobial. Okay, the terminology you might find it called polymicrobial. And so this is an example. So this is the aggregation of cells, and often many species into a complex structure on solid surface. So again, this one I would say a little bit biased. They say called solid surface. Okay, yeah, that biofilm formation cannot happen in liquid. Okay, but sometimes they can break away, and under certain conditions they may form it. But the solid surface is pretty much a kind of like a standard practice that microorganisms get together, they form a community of organisms, and solid surface is the primary requirement, right? And so here you can see this. One of the aspects of this picture, I liked it, is that if you look at these organisms, different organisms, and then can you see this group? It's like a channel, okay? And this is beautiful. I think as you talk more and more, you'll find out the biofilms are designed in a way that these organisms can survive, they can protect, and also they can transport nutrients through the system. It's called channel, right? And so it's kind of an active whole community. They are sitting in an environment. It's like, think about it. If you establish a village, right? Think about it. First establishing a village or community, you need to have a roads, right? To go. You need to have a water source. You need to have a food source. All of those components needs to be in that environment. If you use that analogy, think microorganisms also need that environment, right? They need to be in a community. They need to make that community. That make sure the community has access to water, the food, right? And they can, transport materials across the whole community so that it benefits the whole community. Getting it? And so microorganisms, that's why they make this type of biofilm. And now we are beginning to understand what really goes on inside the biofilm. Okay, this is also another example. You'll see some of those, scanning electron microscopy pictures. These are also, all varieties of organisms. This is also another two different types of organisms forming biofilm. Okay, so whenever we talk about biofilms, what is the benefit, right? So it's a community of microbes held together by certain matrix, right? We call it extracellular matrix or ECM. This is also called EPS, extracellular polymeric substance, EPS. And usually that EPS polymeric substance is consist of what? Polysaccharides, you need carbohydrate which is very sticky, right? Then you need protein. Protein also part of that. What else? We have also lipids, fat or lipid molecules, and then also eDNA. eDNA, you will see that also, we'll talk more about it. It's called extracellular DNA. Okay, the DNA is one of the components of the cells, right? We all have DNA, but when they make this biofilm, the eDNA play a very important role, and it's become very sticky, extracellular DNA. And so some cells in the community, they die, they release this eDNA, and eDNA become part of the community structure. Okay? Again, we'll talk more about these details later on. It protects from sanitizers. So they form biofilm. If you are trying to kill them, sanitizers, chemicals, drugs, antibiotics, they cannot penetrate. Maybe they will kill the organisms which are on the surface, but not necessarily the organisms which are deep into the community. So the protection, you need that protection from having the biofilm structure. Other things, shelter for pathogens. Think about it. So the biofilm, as I said, is a polymicrobial, right? Different types of organisms can form biofilm. But sometimes this polymicrobial biofilm, not everyone is bad candidate in that community. Some are bad, some are good. Some are there for certain purposes. But this aspect of it, you have to remember that. So whenever the biofilm is formed, sometimes you could find that pathogens like to be in that biofilm. You see this? And that aspect you need to pay attention to. You may isolate a biofilm from a hospital setting or from a patient. You might find, oh, this is organism may not be the one causing disease, but have you looked closely? It might have some pathogens inside that community. So it's a good place for pathogens to be in. So it's a good shelter. Nutrient source. As I said, biofilm themselves can serve as a food. Okay? Because they are making these different components. You know what microbes eat, right? If you know what they eat, we know what we eat, right? What do you have to give them to survive, right? First thing would be carbohydrate, carb, right? They like carb. Okay, simple sugar would be the best choice, right? Glucose, sucrose, fructose, whatever, and they like that, right? So the nutrients, right? And so, as I mentioned, some of those things on the top, the community, they're making these molecules, and a lot of these molecules serve as a food. Okay, as they help them together, but they sometimes also they help them to survive because that's their nutrient source. Okay, what else? Persistence. Okay, biofilm, of course, they survive for a long time, right? It's very difficult to get rid of, and it helps the microorganisms to persist on that environment. Okay, this terminology persister, we'll talk a little bit more. I think some of you graduate students will need to understand more than the undergraduate, but I will try to explain you molecularly what happens. So persister is a phenotype, is that these organisms, if you try to kill them with antibiotics, they are not antibiotic resistant, but they can survive. You get it? Something I said. They are not antibiotic resistant, but they can survive antibiotics. Okay? A certain population of cells, when you try to kill them with antibiotics, maybe all of them are sensitive to antibiotics, right? They are all going to die. But certain population within that community, they are not going to die to that antibiotic. But they convert into called persister cells. They can persist in that environment. Have you noticed that when you go to a doctor for chronic diseases, and they will give you antibiotics, right? But then you just never got cured. You always have that persistent infection. Have you noticed that? Or sometimes if you talk to, there are some diseases, this is very common, okay? There is a microbacterial infection or some of these other ones, even though those organisms are not antibiotic resistant, but they become called persister cell. And this is a new phenotype or phenomenon been described last 15-20 years, and there's a lot of studies going on that. But this is an extremely important. And these persister cells, if you try to kill them immediately, they are not going to respond. You cannot remove them. So they are part of this community, that biofilm community. Everybody clear? Again, we'll spend more time on those. Other aspect, we said that biofilms are resistant to antibiotics. Okay, so you are trying to kill them, and they actually can absorb antibiotics. If they absorb antibiotic, the biofilm, it doesn't reach to the organism. Okay, what happens? What else can happen in the biofilm? What about their status? Do you think they are active as if they are outside in a healthy environment? What do you think? usually they are not that active, metabolically active. What does it mean, metabolically active? If I say this terminology, what does it mean to you? Someone said something, but I couldn't hear you. So metabolically active means what? They can uptake sugar, right? They can convert it into byproducts, right? Whether it's acid or gas or whatever may be, right? Or alcohol or whatever. Right? That's these cells are active, right? So that means they can uptake nutrients. Everybody clear? Metabolically active and convert it into byproduct. If I say metabolically poorly active or inactive, but they are not dead. Are you getting it? So think about the hibernation. You heard about the hibernation? The animals go into hibernation, say, during winter months, right? So they kind of control their metabolic activity. So they don't need the energy. They don't need to get out, right? Snakes do that. A lot of wildlife animals do that, right? They can stay alive, but not metabolically or active. They are not doing anything, right? So with a low amount of food, they can survive extended period of time. So if you think that hibernation, the microorganism essentially they hibernate in that community. So if I want to talk about the antibiotic resistance, what do you need to have in order to kill this organism? Then antibiotic needs to get in, right? But if you are not taking in anything, are you going to be susceptible to antibiotics? Everybody see this picture? So this metabolic activity is very important, okay? And I think that was coming. So I started talking about it because I wanted to ask you, it's low metabolic activity. That means they take less antibiotics. Okay, therefore, you cannot kill them. It's very difficult. Okay. And also they play important role in pathogenesis. So you talk about they will adhere, attach, and then also they can evade immune system. Okay. So these are some of those attributes. You can see this benefit to the microbe. Correct? All right. So since speaking of antibiotic resistance, which is going to be extremely important, we'll be talking about, there is a separate chapter only. As just to give you an introduction on the antibiotic microbial resistance. So you can see all of those outline I provided. They are talking about antimicrobial resistance. So presence of persister cells, we already talked about, right? So persister cells, they are not responding to antibiotics in population. High cell density and quorum sensing. We'll talk about that more in detail. Quorum sensing, you heard about quorum sensing, right? How many of you heard about it, quorum sensing? Raise your hand. Okay, so few of you. So quorum sensing, let's talk about that. How do you talk to each other? Do you talk? You're going to talk, right? What else do you do? Nowadays you text, right? Right? Even if you're sitting next to each other, you text, right? So that's a communication, right? So the quorum sensing here is a microbial communication. They talk to each other. They don't have cell phones, so they can't use they cannot communicate. So what they will do, they will release some small molecule. Okay? As soon as that molecule is released, then you say, oh, he's alive, he's sending me some signal. Okay? And then that microorganism is going to respond to you. Okay? So what is quorum means to you? Quorum, when you say quorum, what it means? Have you done any meeting anywhere, anytime? Like you have a group, club, any of those activities, right? Sometimes you have different types of club, right? And if you say you're athletic club, right? So you have certain members, right? And so what do you do in a club? Say, okay, I need to make a policy decision, right? So okay, from tomorrow or from next month, we are going to meet at this time, right? And so okay, this is approved by the committee. Are you seeing what I'm talking? I'm talking about totally different things. I'm not talking about microbiology. It's a human, leadership interaction. So if you have a faculty meeting or professors meeting or student meeting, right? So we need a certain number of people, right? And that number of people are going to vote on that. And then if you have a certain number of people, they vote and then they will pass that. And then it becomes a policy. So in order to pass that, you need to have a certain minimum number of people, right? And that's called quorum. So when you are creating a meeting or conducting a meeting, you want to have a quorum, means a minimum number of voting members, right? And so here, that same information that we started using in microbiology, we call it quorum sensing. Quorum sensing means the microorganism trying to figure out what is their population in that community. Why do you need to know? Why microbes want to know why do we have this many number of microbes needs to be there in the community? What is the single most reason? Say that loud. Resource, right? What is that resource? Nutrient, right? Outs are there. Food, right? We need food. And so in order for microbes to grow, they need food, right? So if I don't have enough food, do we need to grow or multiply? No. Right? So that's the information. How do I communicate to that other member who is about to divide or reproduce, right? Are you getting it? If you have a limited resource, you don't want many of them to grow, multiply, right? You want to limit their growth. So stop dividing. We don't need you anymore because I don't have food to feed everybody. Okay? That communication is very important. So the cell density, cell mass. Are you getting it? If you have a 5 microgram glucose in a test tube, you add some microorganism bacteria, they started growing, right? And then after some time they're going to stop because the nutrient is going to exhaust. So if they're in a community on a biofilm, if that same scenario happens, you need to talk to the friend, right? Stop multiplying, right? Stop reproducing. That way we all can survive, otherwise we all going to die. That's not the goal, right? We want to survive. And that's when the quorum sensing aspect is very important. So that means the microbes realize that we have a limited resource. That's one of the things. It's stop dividing. And how do you tell them to stop dividing? Well, we've got limited number of food, so they will produce some small molecule. And that molecule is going to bind and tell the other person, okay, I got this message. You already told me there is less amount of food. I'm not going to multiply. I stop. Got it? That's how we are all going to survive. And that molecule is the communication. You see this? The microbes talk to each other. That's called quorum sensing. So 5 of you raised hand. Now how many of you are going to raise hand now? All of you? Great. So that's the whole purpose. Are you still not clear? We'll talk about that again. Okay? It's the quorum sensing. We understand that environment and we need to act accordingly. And the microbes, we thought they are just little microscopic organisms. They are just dumb fellows, but they are not. They are pretty smart. They are extremely smart. Okay? And that's why quorum sensing is very important. So I know I spent a little time there. Hopefully by the time we talk more about it, you will learn a little bit more. Oxygen concentration gradient, right? Metabolic activity, the mechanical aspect of it, right? A lot of these factors can help microbes to become resistant to antibiotics. Because you know that antibiotic resistance is a huge problem, right? We all recognize that in microbes or microbiology, it is an extremely important. So this actually summarizes some of those. And then again, like I said, we're going to talk same thing few times. Okay? If you didn't follow it today, hopefully 2 days later or 3 days later or next week when you talk about antibiotic resistance, you'll get a lot of ideas. Got it? Okay. Not to dwell on too much. So the antibiotic resistance problem, AMR call it, antimicrobial resistance. This is the largest global threat. Predicted mortality rates of about 10 million by 2050. So many people are going to die because of the antibiotic resistance. Okay? So biofilm is one of the major contributors to the resistance. Got it? It's not just the genetic manipulation of the gene, which everybody try to study that. Many people study that, right? But this is the other factor. If they are metabolically inactive, they don't take up the antibiotics, they are not going to die. See this? There are many factors you need to understand. And so it's the a compilation or combination of lot of factors. Multi-drug resistant superbugs, Salmonella, Staphylococcus, extended factor we call ESBLs, that group. These are Acinetobacter, Klebsiella, Mycobacterium, Neisseria. These are all these problems. And then, of course, WHO report there are about 490,000 cases of multi-drug resistant TB. That was in 2016 report. So that means it's a huge issue. Again, you need to understand from the, since we'll be spending a lot of time on biofilm, think about how biofilm contributes to that resistance. Okay. All right. So microbes. Then when you talk about microbial biofilms are the major sources of pathogens in food. That's true. We'll talk about that. So let's go to ABC of microbiology. Okay. I'm going to spend time a little bit where those organisms come from, how is the life, right? So we know that there are three domains of life, right? Bacteria, archaea, eukarya. I'm sure all of you knew that, right? Three domains of life. So the bacteria, archaea, they are major biofilm formers. Also the fungi, which is part of the eukaryote, they are also biofilm formers. And then the parasites, we'll talk about that. So when you talk about biofilm, we are not just going to focus on bacteria. We're going to talk about also fungi, parasites. They are also part of the biofilm forming community. They all also contribute to their individual niche in the host or in the food or in the environment. Okay. So bacterial biofilms, we'll talk about that more, we'll learn about more, right? And if you look at these different types of pathogens come through food, and these are a huge list, right? I'm sure you will be able to recognize some of those organisms. Okay. These are we consider called food-borne pathogens. What we learned about them, that any type of food we eat, I'm talking about human or animal feed, and a lot of these pathogens come through. Okay. And just because my interest is in the food-borne pathogens, I'm going to highlight some of them. Aeromonas. Sorry, water got into my. So Aeromonas hydrophila is a waterborne pathogen. Bacillus anthracis, called anthrax, is also animal called zoonotic pathogen. And Brucella, Campylobacter, Clostridium, Chronobacter, Escherichia coli, Listeria monocytogenes, Mycobacterium paratuberculosis, Salmonella, Shigella, Staphylococcus, Vibrio, and Yersinia. These are some of the common foodborne bacterial pathogens. So when you talk about these foodborne pathogens, let me give you just one example. Again, I will study more. You recognize what is in the left? That's called cantaloupe, right? We have cantaloupe in India. You have like a melon family, right? You know watermelon, right? All those other melon family. We have, yeah. This is cantaloupe. It's very popular. It's the same family called cucurbit family of fruits, right? And you look at the structure outside. If you follow the electron microscopy structure, and it has called a net-like structure outside, and it grows like watermelon, right? Any melon or pumpkin or squash grow outdoor, right? And we are having a lot of problem with this. Okay, I mean US. We are talking about, we had outbreak with of Salmonella with the cantaloupe. We have also huge outbreak with Listeria from cantaloupe. A lot of people died actually just eating cantaloupe like this, and Listeria outbreak. So look at this, how this beautiful sweet fruit become a major concern for health, right? People died for eating cantaloupe. So if you look at that, the structure, you can see this net-like structure, and then there's a valley. So this is the part of the network, and this valley, if you look closely, you can see this structure, the plant-based structure. And what it looks like, it's a pretty rough structure, right? And is that a major source of pathogen in the cantaloupe? Well, if it's outside, right? So let's look at it. There are some of this work from our collaborators at Purdue University. So he studied how Listeria actually colonize or form biofilm on the cantaloupe. Okay, and so this is just 2 hours of exposure. You can see a lot of Listeria bound. Just 2 hours. So that means the environment is perfect for Listeria to colonize. Okay, essentially we're looking at the forming biofilm on the structure. And if you look at it 12 hours, you can see more. And these are the 48 hours. I think you can see clearly this is a huge layer of bacteria sitting there, right? Then again, we don't eat the skin, do we? No. So how do Listeria get into the cantaloupe? So we have bacteria. The outbreak the way happened, and there's a lot of research happened. And so everybody's trying to figure out why this cantaloupe become a major source of Listeria nowadays. Of course, Listeria, just to let you know, these are environmental organisms. You'll find them in the soil or plant material. This comes from animals, and these organisms are already there. So if the cantaloupe is growing, if the conditions right, remember I mentioned a little bit about climate change, temperature rise, all of those factors play an important role. More and more outbreak we are going to see. There's no end to it right now, the way everything is going. So you guys have to prepare well to prevent from getting killed or die, right? A lot of people die from this type of just eating cantaloupe. So you don't want to be killed, right? So let's use your common sense. The organisms outside, nobody eats the skin, but how do these bacteria get inside, right? Usually we try to clean it, wash it, use chlorine water, ozonated water, a lot of this disinfectants we use to clean the surface, but a lot of times they could be still there, right? So how do you make a this is how you cut it? You use a knife. What are you doing this by doing this process? It's extremely common sense. Tell me what is happening. You use a knife. So what you are doing actually, if you have a biofilm sitting outside, right, and using knife, so knife is helping the microbes to eat. You see, that's very simple practice. You don't think about it. Any fruit, I'm giving this as an example. Fruit, you cut, the microorganisms are already outside, right? And when you cut it, these organisms are going to get in. And what happens? If you cut inside, you already know the inside is very juicy, right? It's rich in sugar. Who likes sugar? Microbes, besides us. They like sugar. Anytime, anywhere you give some sugar, they are going to grow, right? That's their favorite, and they are going to grow. And so when you cut it and living there on the room temperature for a few hours, like this type of weather, oh, they are going to just multiply so rapidly. Okay, so most of the problems we have when you buy this cut fruit, okay, and you need to take a lot of precautions to prevent it. So can you see this, how the biofilm contributing to people's death or suffering? You see this one? So this is a direct example. And so they are trying to find a different way to remove this biofilm. All those different strategies. We'll talk about some of them, okay? Use some food sample, and then how do you remove this biofilm? That way you can prevent it. What about yeast and mold? We talked about bacteria, yeast and mold, they also form biofilm, right? Look at this. These are Aspergillus, Candida, Coccidioides, Cryptococcus, Mucor, Penicillium saccharomyces is the yeast. So these also can form biofilm. And this is a classic. We had a tomato in our basement. We left it for a while. I forgot to, bring it and eat it. And then I went there and downstairs. Usually I don't check it. And I went and picked up this tomato. Look at this. What happened? All the mold growing, right? Mold is a good spoilage organism. And if you think about, that's a beautiful biofilm, right? They form a nice structure. So if you look at the yeast and mold, these are the common structure of the yeast, the mold. And you have seen this, the mold species, Aspergillus, Penicillium, and they make mycotoxin, okay? And mycotoxin is a deadly toxin. It causes cancer. Okay, a lot of times you eat certain food, and if you continue to consume mycotoxins, there are chances that you are going to get cancer. Especially the Aspergillus toxin or aflatoxin, which is a type 1 carcinogenic agent. How many of you thinking that eating food can give me cancer? We don't link that, right? Think about if you continue to eat mycotoxin tainted food, like a peanut, a lot of those nuts are a favorite place for a lot of mycotoxins. Okay, so there is a certain standard. You try to monitor it before you consume it. So this is an example. They can grow, they can produce toxin, and that mycotoxin you cannot heat inactivate it. You can cook it many times, you can still cannot inactivate mycotoxin. Okay, there are special way you can inactivate it, but very rare. But if consuming mycotoxin built up in your system, it can cause you skin cancer, mostly the liver cancer, hepatocarcinoma, also the kidney cancer. Okay, and all of those starts with the mold growing, forming biofilm, and producing toxin. This is the picture of a yeast on the left, and they also form biofilm. Okay, biofilm produced by Saccharomyces on the left, and the Candida albicans, actually it causes disease, serious disease. Okay, it's a mold-induced disease, and they are also notorious biofilm formers, big biofilm formers. We'll talk about some of the clinical disease. Then we already talked about that one, the biofilm. The parasites. Okay, think about parasites. We ignore that parasitic diseases, but not in India. I know we have a lot of parasitic diseases because of the higher temperature, right? And the tropical countries, they suffer from a lot of parasitic diseases. Okay, so we are not talking about metazoan parasites. We are talking about unicellular parasites, means single cell parasite, and those are like Cryptococcus, Cryptosporidium, Cyclospora, then Entamoeba, Giardia, Isospora, Toxoplasma, Leishmania. These are single cell or unicellular parasites. Okay, and they also form biofilms, and they become a big part of the problem. So there are many diseases. We'll talk about that. How are we doing with the time? Probably he's not keeping track of. 12:30. We'll finish at 12:30 or what? What time are we supposed to finish? 12:30. That's okay. I don't want to, because we want to stay on schedule. So let me finish the first part of the introduction. This is an example of the unicellular parasites. So we're looking at Giardia, Entamoeba, and this is Toxoplasma. These are the unicellular organisms, okay, and they form these parasites, the biofilm. We'll talk about some of those as we go along, the diseases caused by these type of organisms. You heard about tapeworm, right? Tapeworm is not unicellular. This is metazoan, large parasites, and people suffer from, especially unhygienic conditions. Tapeworm is kind of dangerous. Then also a roundworm, those are the long ones, but they don't form biofilm. These are like large ones. But I'm talking mostly about the unicellular on the top. Okay, so how actually the biofilm help the parasites? Okay, unicellular parasites. So if you look at there are three functions I listed. So parasites, and they can sometimes destroy the biofilm, okay, because they make enzyme. They produce gastric the enzymes, and that enzyme actually can destroy biofilm if the biofilm is produced by bacteria. That's number one function. Number two, parasites influence biofilm formation. So some of the products they make that can help biofilm formation. And then the last one, biofilm can protect parasites. So if there is a biofilm formed by bacteria, and then parasites can be hidden. Remember I was mentioning before that it's a shelter for many pathogens. They can be all in one environment. And so parasites, and they can definitely survive inside this bacterial biofilm, and then eventually that can play an important role in causing disease. Okay? I want to point out that every slide, most of the slides has certain references, and these are all recent references. If you are interested in learning more, always you can go to that article and find more information. Okay, keep that in mind. Okay, so this slide. I think this is talking about fungal biofilm. Okay, and what they are looking at is that there is a huge application of biofilm in the industry. And so if you look at that, biofilm of fungi helps citrus resist acid rot. This is kind of interesting. That's the reason I put it up there. We talked about parasite, but this one mostly for the fungi. I should have switched the slide before. Biofilm was attached to the water pipe. So they created water pipe, that happens. Biofilm accelerates the corrosion of food processing equipment. So the application in a sense both negative and positive. Okay, it's saying that, well, if you have a biofilm, we know that that's going to corrode equipment. Okay, because of the way they behave, they eat up the equipment, right? And like I showed you the pipe, biofilm contamination of implanted medical device. That's of course a huge problem. We'll talk about that. Biofilm production generates secondary metabolites. Definitely that does. Biofilms are used to feed broiler chicken. They use biofilm to feed the animal. Biofilm are used to treat sewage. Biofilm are used for biological as an alternative to pest and disease drugs. So huge applications you will see with the different types of biofilm. Let me see. Maybe this is a good place to stop. Then we'll talk about biofilm. One of the things I want to send you the message, like saying that not always bad. Okay, so biofilm, if you look at the beneficial, foliage and pathogenic. So to me, I think the beneficial biofilm are helpful. We need biofilm to be produced, can help certain diseases, not diseases, we should not talk about disease, certain fermentation process, okay, certain gut health or improve health, you need biofilm formation characteristics. Foliage also part of that. I showed you some picture, they can spoil food. That affects the sustainability aspect, if the food spoiled by biofilm formation, and then of course the pathogenic activity. Okay, so we've got some slides, we'll talk about that. And then I know today is the first day, probably everything time wise, but hopefully from tomorrow we'll be on time, on schedule. And then again, make sure when you go home, or home means your residence where you are staying, make sure go over these slides. You have a good familiarity with these slides. That way next day when I come and talk about it, so that you have a good idea what I'm talking. Okay? And I need that feedback. Otherwise, your learning won't be heightened. We need that you go through these slides, even if you don't understand any of those slides, that tomorrow's lecture, but just go over that. Okay? It gives you familiarity. And that way when we talk, you will have a better grasp of material. Got it? So I think I'm going to stop here. And then I think you have a lunch break, right? Okay. And then after that, Dr. Shukla going to talk. Okay. Any questions before I go? You should have a lot of questions. Again, prepare bring questions. I want to see your learning. Because if this is more my way of teaching is that I don't want to keep on talking, and then you will be sitting there listening and not really understanding, or you don't want to ask questions. Don't feel bad about questions. Okay, I never get offended by any question. There is no question called stupid question. You knew that? You heard about it? Stupid question. People are asking stupid questions, and it may be a stupid question. I shouldn't ask it, right? And that prevents you from asking the question. But there is nothing like that. There is no question is called stupid question. You heard about it? So questions are always good. Any questions? No. That's why you have to ask the question now. You ask me now. I will ask you later, right? Or you are going to ask you later. Go ahead. Oh, okay. We'll talk about that a little bit more. eDNA, just to simply, what I was trying to mention, that DNA molecule is very sticky. Okay, if you break open a cell, the DNA gets out. They are extremely sticky, and so when this in the community, some cells die, some remain alive, and their DNA gets out. Okay, and that eDNA, which is called extracellular DNA. It's kind of funny, right? E stands for not electronic, it's extracellular. Everything nowadays E, right? So this is extracellular DNA comes out and it makes it sticky, so it helps the biofilm to stay together. And that's what it is. And the DNA molecule in general, they are very sticky. Okay, the type of skin infection we get, which organism cause most of the skin infection? Anybody said Staph aureus? We're going to talk more. Carbuncle, boil, right? Out of the skin is the toughest place, right? I usually use this example. Staph aureus makes a lot of nuclease enzyme, tons of. The reason they do it, one of the things is that as soon as they make this boil or carbuncle, you have the skin infection, right? Usually come from staph, and you squeeze it, the white pus come out. You know what I'm talking about, right? We all suffer from. This is nothing new. If you are a little bit not taking shower frequently and all kinds of stuff, your skin, all are inhabited by Staph aureus, and skin infection is very common. I know the mold, they also can cause it. So the Staph aureus, the reason I brought this up, they make a lot of nuclease. And the reason they make a lot of nuclease, because as soon as the infection happens and this DNA gets out, that can impede the progression of the disease. So that's why they need to make the nuclease to liquefy the DNA. So when you squeeze it, you will see all of those DNA are no longer fresh DNA. It's a lot of dead immune cells, a lot of dead cells, and then already bacteria can infect much easily. Because if you break open one cell, the DNA is nasty. That I know I gave you a long answer, but the whole idea being is that this is a very sticky molecule that helps the DNA, the biofilm formation. Okay. Any other questions? Yeah, go ahead. I think you have to speak louder. How benefit or? Oh, for treatment. What treatment? Oh, sewage treatment. Oh, I see. That's a very good point, though. Just simply, I can tell you that the biofilm being there and producing enzymes. That's a good point, though. I don't know. I have the answer. I'll find out that answer. The sewage application. I think it will be like a lot of enzyme production, and enzyme production may help clarify certain molecules. This is the environmental side. A little bit less familiar. You may have that answer, but I think I'm thinking more like a fermentation process I will talk about, and there is a biofilm formation, help fermentation, for example, but I'm thinking more like a byproduct production. The organisms being there, producing certain enzyme, are they clarifying certain molecules from the sewage, removing certain, undesirable molecules. Okay, if you don't have any other questions, then we'll go for the next page. All right, I'll see you. I think I'm done for today, but then we'll start tomorrow morning.
Lecture or not. Just quick question. Can you tell anybody where do you find biofilms? Anybody? No? Any solid surface. Actually partially true, not completely true, but loudly. Okay. The correct answer is biofilms is almost everywhere. Many surfaces, wherever you find moisture, up to some extent, some kind of biofilm is always there. You may have seen many biofilms by naked eye, like on rocks, if you may see, especially in rainy season, if you see it becomes slippery. That is nothing but the biofilm. In your washroom, bathroom, if you leave water in bucket for some time, and the next day you will see that some slimy thing is there or some sticky thing is there. That is nothing but the biofilm that you can feel by naked eyes, or you can just touch. You will feel that okay, something is there. Over the period of time, this was realized that biofilm is almost universal. Almost all the biofilms, all the bacteria, up to some extent, they are present in in a way of which is called biofilm lifestyle. And you know that many bacteria, or most of them, actually are sessile. Sessile means which are not able to move. Okay, and only very small fraction of them is known as planktonic. Planktonic means which can move in the water body, or from one place to other place it can move. That is known as a planktonic one. And from this planktonic also, only small fraction of bacteria is known as culturable bacteria. Like you take water sample, you put it on some growth media, you will see next day you will see some colonies. But it is not actually representing all the bacteria which is present in the water or from wherever you have taken a sample. Why? Because it is estimated like 95% of the bacteria, they are unculturable. You cannot culture them. So whatever you see in your lab is very small fraction of the bacteria which is actually present in the environment. So as I said, bacteria live in two major forms. One is planktonic. Planktonic, when I say, so they have some kind of structures which help it to move, like flagella or some pili and all. So, and the second lifestyle is known as sometimes it is known as a colony. When you see on the plate, that is colony, mat, flock sometimes, and biofilm. It is sorry mistake. So it can be some bacteria which are not motile, but they can relieve as isolation, like Staphylococcus. It is not a planktonic one. It cannot move, but it can live in a free form. So that is called as like it is live in isolation, or it can live in association. On the other side, which are able to move, planktonic one, it can remain as a planktonic, or it can become sessile. Sessile lifestyle is biofilm, and planktonic or isolated one is this lifestyle. So as I said, it is a almost universal feature, and people believe that it is a universal feature of all bacteria. It may form on solid substratum in contact with moisture, and soft tissue surface, like you have an infection on your skin or any bone, that is a soft tissue, and liquid interface. When you try to grow many bacteria in the your lab, you will see all bacteria, means all the time it is not needed that biofilm is present on the solid surface. It can even form on the liquid air interface. I will show you how does it look like. So this biofilm, here you will see this kind of biofilm. It is not on any solid surface. It is between air and liquid. And you have different other one which is solid surface attached biofilm, which forms at the bottom of the vessel whatever you are using for your biofilm growth. And if you see its microstructure, this is like whatever you can see by your naked eye. But if you look at the at the microscopic level, they may have this kind of structures. What Professor Bunia was showing, it may have a different complex structure. It may have a mushroom kind of structure. Their water channels will be there through which many nutrient or if you give some treatment, it will pass through that water channels. They are called mushroom kind of biofilms. Let's come to the biofilm, how it grows, and what is the normal lifestyle of a bacteria. So it is, let us start with the free cell of any bacteria. Sometimes it is stress, but not all the time. Sometimes it is a different factors which initiates the biofilm formation. Like if they find some surface which is good for attachment. What they do, they will change their genetic level expression or phenotypic changes will be there. Then there will be attachment. This attachment again divided in two parts, irreversible and reversible. First it is reversible, like the first contact they try to sense whether this surface is good enough for the settlement. If it is good enough, then it further move on and becomes irreversible by developing some mechanism. Then after this attachment, then it grows in form of monolayer, and then other sensing like quorum sensing or some gene expression changes will be there, and some gene will be upregulated, two genes will be downregulated, which will be in the courses you will see, because I have seen the list. Definitely this will be covered, that what kind of genes are involved in the upregulation and downregulation, where it transforms from the free cell to biofilm. Then there is a microcolony formation, and then if things are good, it grows into form of a mature biofilm. At this stage it is known as a mature biofilm. There you will see that kind of structure what we are talking about, like mushroom structure or water channels, etc., etc. And in some adverse effect, or sometimes you treat with something, or if they find that nutrients are not available for the further growth, they may detach themselves. Different bacteria have different mechanisms for this thing. And again it becomes, you may say planktonic, mostly planktonic term is used for this detached cell, but it is not exactly same. I will come to that part. And here I am showing you a good picture of a scanning electron microscopy, where you can see how a microscopic level how a biofilm is look like. Here you have small that you can see dot like that, those are bacterial cells, and it is cemented through EPS which has carbohydrate, protein, and so many other things. And you can easily understand how it is looking, that once they form biofilm, they have an advantage as compared to free cell. It provides a physical barrier. Physical barrier means when you are treating with some antimicrobial, antibiotics, the first line of defense is that you have a physical barrier. Those antibiotics has to pass through that EPS to reach the cell, then only they will be able to show its impact. Dormant lifestyle. What is the meaning of dormant lifestyle? It is different from the persister cells. Dormant lifestyle means, as sir was telling, that when cells are metabolically active, then at that moment, if you are giving any antimicrobial, particularly antibiotics, they are more effective. Why? Simple reason is, mechanism of antibiotics, right? Anybody? Antibiotics? How many people know what is antibiotics? Any example? Example of antibiotics? Sorry? Speak loudly. Gentamicin. How does it work? It targets something, right? What? Protein machinery, right? So when cells are metabolically active, they have a lot of protein synthesis, DNA replication. Okay, if you see a typical bacterial growth curve, you have log phase, where cells divide, a lot of protein is synthesized. That time if you add any antimicrobial, it will be much more effective, because it will target protein machinery, DNA replication, right? Or ampicillin, which target cell wall formation. But if all those things are not happening, means metabolically very low profile if they are, that is called dormant lifestyle, because they are not very actively involved in the protein synthesis or DNA replication. All those whatever antibiotics you are giving is comparatively less effective as compared to planktonic one. Then comes to persister cell formation. Persister cell is one step below of dormant lifestyle. Dormant lifestyle means you have a low metabolic rate. Persister cell is like coma for the cell. Coma means it is just life, just very basal expression of all the things which is needed to just to survive, nothing else. Almost zero your protein synthesis and all. So all your antibiotics and all is no more effective. And that is why after a while, once like whenever you go to hospital and get antibiotic treatment, nowadays doctor always tell that complete the course. Is it not? Why? Because once you take antibiotics, most of the actively dividing cells will be killed, but some dormant lifestyle bacteria or persistent cell will be there still. Once you stop, again they will become active and again you have infection. That is why they say that you complete the course for 3 days or 4 days, because once actively dividing cells are killed, these persistent cells will grow. When you are taking second dose, third dose, those will be also killed. Then only you have complete eradication of the infection. Otherwise, it is very difficult. And these are the things which are contributing to antibiotic resistance and persistent infection. As I was saying, when it is dispersing, it is not exactly planktonic cell. You cannot say, because there are still some differences. Let's see, planktonic homogeneous population, single cell specific substrate, means they utilize high metabolism, high motility as well, low attachment and antibiotic susceptibility, low virulence. But if you see dispersed cell, they are not the same. Planktonic cell means all bacteria are almost same, but in the dispersed cell, which is just dispersed from the biofilm, you have a phenotypic diversity. Phenotypic diversity means few are completely just like planktonic cell, few are still in the transition phase, which are switching from the biofilm lifestyle to planktonic, and few are like just dispersed but still not changed. So here you have phenotypic diversity, and they have high metabolism like planktonic, but because just now it is coming out of the biofilm, still it has antibiotic resistance associated. So you can see it is not exactly same, they look same, but they have some differences. So these are the major bacteria which is being used in most of the labs for study of the biofilm, and most of the people, they their work is mostly around bacterial signaling, why antibiotics are not very effective, how gene transfer happens, horizontal gene transfer, because of which you have MDR problem. So let's understand what biofilm matrix is made of. You have seen that you have biofilm, you have cells, and it is surrounded by some EPS. That EPS is common term is biofilm matrix. Why matrix? Because it is kind of black box. There are so many things. The more you read, more you will become confused. Really, you will not understand that what exact mechanism is, because for one bacteria something is happening, but if you think that same thing will happen with the other bacteria, it is not like that. That is why it is also called a black box of biofilm. So let us see what are the major component of most of the biofilms. Polysaccharide, which is almost common for most of the bacteria. Then water is the major part, because if you see only living cell, any cell, 90% is water, right? Same goes with biofilm as well, 90% is water. You remember this thing because it helps in many other understanding of the biofilm, that it is 90% water. Then you have surface protein. You have two kinds of protein which is involved. One is surface protein. Surface protein means one part of protein is still attached with the cell wall of the bacteria, and then you have amyloids. Amyloids are not attached with the cell wall. Mostly this protein is exported outside of the cell, and they form a because of hydrophobic interaction, they form a very it is very difficult to even degrade by using any protease. That is called amyloid. Then you have biosurfactant. Biosurfactant is used in regulation of the biofilm. At the last stage you saw that there is a dispersion of the biofilm. It is not always from the outside. Bacteria itself, they decide that we need to move away from this place. So that time they produce biosurfactant. Up to some extent dispersion happens because of biosurfactant. eDNA, extracellular DNA, Professor Arun already told, extracellular DNA, it comes from the bacteria itself. Then lipid. Okay, and then comes the calcium. How calcium can help? Any idea how, why calcium is involved? Simple biophysics, enzymatic factor, sometimes but not majorly. Yeah, that is correct up to some extent, but again it is not always the case. I will show you example. Simple biomechanics, you understand, it is positively charged. Polysaccharide, it is negatively charged. eDNA, if you see, again it is negatively charged because of the phosphate backbone of DNA, which is negatively charged. So what it does, it interlinks everything. It provides a lot of stability. So these are the individual components, and each component always not participate in only one function. Each component can participate in many kinds of functions. You can see the polysaccharides are involved in so many things, even retention in the water because of having hydrophilic residues, and sink for excess energy. This you try to understand. Why biofilm is a preferred mode of life is that it is like whatever they are getting, like the way we do, we'll eat whatever extra there, that we will keep in the fridge, right? And whenever we are hungry again, that we will take and eat. Same thing goes for the EPS. So whatever extra they are getting, they are storing in the form of EPS, protein or polysaccharide. So when nutrient is not coming from the outside, they will use it. Or in normal, like natural mode, whatever extra they are getting, because it is not always available in the water, right? So they will store it in form of polysaccharide, and whenever it is needed, they will use it. Proteins, enzymatic activity, especially when dispersion has to happen, that time it is involved. Adhesion and cohesion, and this also thing for excess energy, but it is secondary. Polysaccharide is the primary one. And divalent ion, as I told you, mechanical stability, regulation of biofilm associated protein, that I will show you now. And if you see one by one, we will talk. Adhesion, for adhesion, mostly polysaccharide proteins, DNA and amphiphilic molecules. For adhesion, whatever mostly we have discussed, everything takes more or less part of in that function. And protective barrier, I already told you, absorption of organic compounds and inorganic compound as well. Enzymatic activity, especially digestion of exogenous microbial molecule, like I told that it is stored in the form of polysaccharides, but polysaccharide it cannot use. Whenever they need to use it, they have to be broken down in sugar monomers, then only. So for that thing you need enzymatic activity. And as a nutrient source, potentially all EPS component, but primarily exopolysaccharide. Exchange of genetic information, that is genetic information means only DNA can participate. Biofilms are extremely good for this purpose because they hold two bacteria together very close to each other. You must have read in like transformation, conjugation. Conjugation is one way, where with that through tube it passes the DNA. If they are in the form of biofilm, because all bacteria cannot form that FPL, that structure, right? But biofilm, when it is in the form of biofilm, there are two mechanisms, because two bacteria are close to each other. That is one thing. Second thing is, sometimes bacteria die, and whatever DNA it has, it is secreted outside, means it is burst out, and that DNA can be taken up by the nearby bacteria. That is how you have a genetic information transfer, that is called horizontally gene transfer. And yeah, electron donor acceptor in some particular case, which is done by the redox proteins. And sink for excess energy, okay, same thing. Yeah, okay. So let us see one by one. Exopolysaccharide, any polysaccharide, a polymer of polysaccharide you get from the monomers, right? Glucose, then you have a layer of exopolysaccharide. Then different kind of exopolysaccharides form a network of polysaccharides, and these polysaccharides, they have both kind of group, hydrophilic group and hydrophobic group. and these groups can participate in different kind of functions. Okay. Let's see how biofilm associated proteins are involved. Even though since people know about the biofilm, it was always believed that it is polysaccharide because that binds cells together. But later on, first time in 2001, one protein is reported that is known as BAP, biofilm associated protein. It was shown that if you this is present in the S or S. So once you delete this protein, its capacity to form biofilm significantly comes out. That means there is some protein which is involved, and it is not only in the primary adhesion. Primary adhesion means that initial part of the biofilm formation, but they have a different kinds of protein which forms give different kind of functional capacity to the biofilm. You can see here in this schematic picture that blue color is the bacterial cell, and this green one, which is you can see at the bottom. Okay, so this one is TAS A protein actually, which is involved in the primary adhesion. So this is from the bacillus. So if bacillus wants to form biofilm, it will first produce the TAS A, which will form a layer on the surface. After that, bacteria comes, sits there, then it will start producing different kind of protein, and which have cell to cell interaction. So you have different protein for the attachment to the surface, and if they want to interact with each other, they have different set of proteins. So different surface proteins or proteins you say is involved in different kind of functions. So after that discovery of the first BAP protein, which was in Staphylococcus, people discovered so many other biofilm associated protein, and most of them have a different kind of protein, but they share some common structure. Like for many other protein, let's say any enzyme, okay, any protease if you take, whatever protease we have in our gut, or if you take from a buffalo or from cow, and if you sequence it, you will find that it has a lot of similarity. Almost 90% will be 99% will be same. But these proteins, even though they are performing similar function, they have entirely different sequences. They have some common structural similarity, but not the sequential similarity. I will come to that part. Okay, now come to this part, that as I told that they are, even though they are ubiquitous, but nature of biofilm is bacteria to bacteria varies, but within same bacteria also it varies. Or what I mean to say is, if you take example of S. aureus itself, forget about other gram negative or other bacteria, only gram positive bacteria, if you see, the first those bacteria which were discovered for S. aureus, it was found that they form a lot of polysaccharide. Then when BAP was discovered, in that it was found that majorly proteins are involved, and along with that, along with the protein, eDNA is also involved. So if you look at this structure, you will find that they are differently, they are very different from each other. Here it is dominant by the exopolysaccharide, and here you have a protein dominant and eDNA dominant biofilm. Obviously, if you want to deal with this biofilm, because their nature is different, you have to have a different strategy. Okay, we'll come to that part later. And then you have third kind of S. aureus biofilm, where PSM fibers, which is phenol soluble module fibers, they are again different from these two. So what is the significance of having different kind of biofilm? Significance is predominantly you can understand in the case of methicillin resistance. Methicillin resistance is one bacteria where most of the penicillin derived in antibiotics are not effective. Methicillin, which is very advanced version of penicillin, also not effective. You see this one is methicillin resistant, and this one is methicillin sensitive S. aureus. And you see this is sensitive one is dominant in exopolysaccharide. And if you see the same image, you can see how it is looking like. It is like looking like a clump because a lot of polysaccharide is there. But if you see the MRSA, methicillin resistant one, because here proteins are dominant, which is small molecules, you cannot visualize directly here, but you can see that it doesn't have that kind of cementing structure, right? So they are very different, and if you want to deal with them, you have to deal in a different manner. Okay. So there are various methods to regulate the biofilm. So here you have free living bacteria, and here you have a different form of bacteria. So it depends on, as I told, that it depends on many properties. One is physical properties of surface. When it comes and senses what kind of surface it is, as I told you, those factors are how rough it is, what is the topography, what is the binding energy for that surface, and hydrophobicity. These are the major factors. Of course, many factors are there, but these are the major ones. Simply, you can, in simple layman terms, you can understand, if you want to grow biofilm on a very rough surface, it is easy. Okay, bacteria can easily get attached to that one. But if you have very smooth surface, let's say like smooth glass, there it may form, but it may not be stable because it can easily be sloughed off. Then apart from the physical property, chemical properties as well. Okay, how chemical properties, like hydrophobicity I told, like in our lab itself, we want to develop anti-biofilm surfaces. Okay, what we do, we modify the hydrophobicity of the surface and see how bacteria is responding, mainly marine bacteria, how they are responding to the towards the biofilm formation. And mostly if you have higher hydrophobicity, you will have a lesser biofilm formation. So chemical property means sometimes if you have functional groups available on the surface, it becomes easy for bacteria. It can have covalent linkage with those surface, but if it is a chemically neutral surface, then it becomes difficult. Chemically neutral means like if you say this one Teflon, which is used in the, it doesn't have any very active surface. It doesn't have any active functional group on the surface. So bacteria find very difficult to form biofilm on such kind of surface. Then comes environmental factor and genetic factor. Environmental factor means what is the temperature, what is the pH of the that microenvironment, and how much nutrient is present, other things. Genetic factor means what is the inherent capacity of that bacteria to form biofilm. So these are the genetic factors, that how much exopolysaccharide production ability it has, and there is some secondary messenger to regulate production of biofilm matrix. And one is well known, cyclic di-GMP. I'll just give one very brief idea what is cyclic di-GMP messaging. And a group of surface protein, as I told you, which is known as a biofilm associated protein. So this is, as I told, that most of the biofilm associated protein, they don't share the similar sequence, but they share a similar structural property. What is that structural property? You can see that this biofilm which was first discovered, you can see the size, it is 2276 amino acid long. If you know what is the normal size of any protein, you know what is the size, hemoglobin? No, but anyway, it is not that long. Mostly it is 50 to 100 amino acid long, most of the protein, but it is 2200. And if you see its molecular weight, it is 238 kilodalton. It is very, very huge. Normally protein will have from very small to 40 kilodalton or 50, 60, maximum 70, but this is 238 kilodalton, very huge protein. What kind of structure they have? You see, whatever I am showing here as a red one is calcium binding motifs. So this protein has 4 calcium binding motifs, and there is a presence of tandem repeats. So in one domain, mostly at the C terminal site, it will have tandem repeat. Tandem repeat means it will have same sequence in a repeated way. It has 86 amino acids which is repeated 13 times. So what it does that it mediates the cell adhesion. Protein to protein interaction will be there, and that's how it promotes the biofilm formation. There is several theories that how it works. It may attach to the surface of the other bacteria, like in this first one, or protein-protein interaction may be there, or it may intertwine with each other, and that's how it can help in the biofilm. But one thing is for sure, that if proteins are involved, as I showed you in MRSA, if you target protein, it will be easier to deal with such kind of biofilm. I will give you one very brief example. I will not go into much detail. I am talking about this BAP protein. You have two strain. One is BAP positive, BAP negative. BAP negative means where everything is same, but only BAP is mutated. So if you see this one, in this first one, as you increase the calcium concentration, biofilm formation is coming down. So as I told you that you told that calcium is mostly increase the integrity of the biofilm. Most of the time it is true, but here it is not true. If you add calcium here, it will actually bring down the biofilm formation. And it is not that it is killing the cells. Here you can see, if you see the planktonic growth, it is almost same in the presence or absence of the calcium. So what it is doing? It is regulating the protein. It is binding to the protein, it is altering its structure, and somehow it is making it non-functional. You can see if you grow this biofilm in the presence of calcium and in absence of calcium, you can see striking different kind of biofilm. Okay. Then what we can do, as I said, that if they are participating in the biofilm formation, why not to divide them? Why not to attack BAP? So what we do, what we did, we treat with the proteinase K. Okay. And mind it, I am not showing other one, but this proteinase K treatment we tried with the other kind of SRS where polysaccharide is dominant. There you will not see the eradication of the biofilm. But if you treat this biofilm in the presence of proteinase K, you will see that there is a removal of the biofilm. We have a lot of bacteria which are protein dominant, and most of the case you will see there is a decrease in the formation. Inhibition as well as dispersion. Dispersion means once you have a biofilm grown and then you treat, you call it biofilm dispersion. And what is the main benefit of this strategy? Here I am showing you this red one is, let's say, X concentration of antibiotics, and green one is 5 times increase in the antibiotic. You can see in all the case, you increase the antibiotic concentration, it is not helpful. You can clearly see that 1x or 5x, killing is almost same. Why? Because as I told you, and it provides a physical barrier. Antibiotics become ineffective. But what happens with the same concentration? You just add proteinase K. Okay, what it will do? It will cleave all the proteins which is binding each other. Your cells will be free. Now again it becomes susceptible to the antibiotics. So instead of increasing the concentration, you just mix with the proteinase K or any protease, some other protease. It will become again sensitive to the antibiotics. And it was found in many other strain as well, this kind of observation. So this is that some in silico analysis that we did. This part is that repeated structure which I was telling you. Okay. So here you have random coils kind of structure, but this part where you have calcium binding motifs, here you have different kind of structure. Okay. So we wanted to know that what is the purpose of having this repeated structure. There should be some reason why, because if you see from bioenergetics point of view, if bacteria is secreting such a huge protein, it is wastage of resource if it is not serving any purpose. Correct. So if it is having such a large C motif, it may be serving some purpose. That's why it is retaining it. Otherwise, why it would? So this one we did some in silico analysis, but here you have a patch, STVTVT, this patch, amino acid patch, using different kinds of tool, which is not useful. But anyway, what it looks for is whether this part of the protein, whether it will form aggregate based on its hydrophobic nature. So all the tools suggest that it has a high hydrophobicity. That means, and again we some other antibiotic, in silico analysis, just to make sure that where this part will be present, it shows that it is present on the beta sheet. So beta sheet is here, okay, here, this part it is present. That means each repeat has a globular kind of structure, and here you have one hydrophobic patch, here you have hydrophobic patch. Once it is present in the aqueous solution, what tendency it will have? It will have tendency to mark up, right? A hydrophobic patch will come together, and that's how you have a cell to cell interaction. Yeah, so here it is, okay. So here, so what we thought, like we do in PCR and all, if we know that this part is participating in two global interaction, if you mark it, you put something which covers this part, can be used as a inhibitor. So it's very simple logic, because STVT, that heptapeptide present in this one and in the other protein, they are interacting. So what we will do, that will provide an extra this heptapeptide, which will go and bind here. So this part will not be available to bind to other protein, right? Very simple, and it is not having any killing activity. But we see that it is bringing down the biofilm growth, right? So as I said, that if you understand what is the mechanism, then you can devise some strategy how to deal that particular biofilm. So this is going on anyway in our lab. That as we know that it is anyway, it is because the hydrophobic patch, if you want to solubilize, it is not easy. If you put in water, it will not be soluble, right? Because it is hydrophobic. And if you want to use it for some purpose, like in clinical application, it has to be up to some extent it should be soluble in the aqueous solution. What we do? What we are doing, we are trying to modify it little bit by adding some different different amino acid, positive or negative, so that it has, when you add any charged amino acid, what will happen? Its hydrophilicity will increase, right? But at the same time, it should not bring down its ability to bind, right? So this is the way how we find out. We have a different kind of a structure, then we see what should be the binding affinity. More the binding affinity means it will bind better, okay. So these are the how you first do the bioinformatics and predict something, instead of having all kinds of possible thing and getting it synthesized, because it will be wastage of money. First we try to understand whether it will work or not, because so many tools are available nowadays. So better you use them, and then if you see some positive outcome, then you test in wet lab. So we saw that okay, this one is showing very close to the original one. This is minus 5.6 and this is showing 5.7. So we are getting it synthesized. Maybe in couple of weeks we'll see whether it is really working. Okay. Come to the some messenger which regulate the production in the biofilm. The most common messenger which affect the biofilm formation is quorum sensing molecules. In the first few slides I was showing, from one stage to second, third stage, it is shifting because of the quorum sensing molecules. And quorum sensing molecules, what it is actually, it is a way of communication between bacteria. This is how they talk to each other. You must have read that common bacterial growth curve. You remember it is S shape. Okay, that is lag phase, then it goes into log phase, then it starts declining, then you reach to stationary phase. The question is, why bacteria reach to the stationary phase? In nature you don't have that much nutrient, we can understand, but in lab we have lot of whatever growth media we use that has a lot of nutrients. Why it reaches to the stationary phase? It should keep on dividing, right? But still after some time it stop dividing. Reason is quorum sensing. Okay, because after some level they understand there is no further need of division, so we should stop, because when they are dividing they are using resource. For each division they need 2 copies of the DNA, right? So they are utilizing resource. So after some point they have to decide that okay, this is enough, we have to stop, and that is how it is done using quorum sensing. So this is similar, I mean very simple depiction of how quorum sensing works in general. Okay, so when they are low in number, they keep on producing some autoinducer which is excreted outside. Okay, so when they are low in number, these molecules which is commonly known as autoinducer, they are also low in number. Okay, but once they reach late log phase, means they are normally in 10 power 8 or 10 power 9 cells per ml, that means very high density. That time each cell, because it is secreting this autoinducer, in media also there is lot of these autoinducer molecules. Okay, once they have very high concentration of autoinducer molecules, they start going inside. Okay, at that point, and then they go and bind. There are different receptors in different bacteria, different receptors, and it goes and stop those genes which are participating in the division. Okay, or many other things like virulence and so many other factors are there, but there they go and stop or change the gene expression pattern for bacteria. So in what kind of, I just told about the growth, but this quorum sensing is involved in the secondary metabolite, exopolysaccharide pigmentation, biofilm, motility, enzymes, virulence, biosurfactant, so many functions are there. So secondary metabolite, you may be knowing that, you must have read in textbook, the secondary metabolite is mostly expressed in the stationary phase. How? Because once quorum sensing is activated, then only means division stops, then these kind of things start happening, means secondary metabolite production or exopolysaccharide when they are entering into the biofilm production phase. So there are different kinds of autoinducers. Okay, and it is different in gram negative, and gram positive has a in general different one. So classical autoinducers are known as a acyl homoserine lactones. Okay, you have homoserine lactone and you have acyl side chain. Acyl side chain can change, it can be 4 carbon to 5, 6, 7, 8 up to 12. You have different range of AHLs, and each AHL is involved in different kind of regulation. Auto-inducing peptide mostly in the gram negative SRS, and then you have auto-inducer 2, which is for the interspecies. These two are intra-species communication, means like if it is Pseudomonas, it will be for the same species regulation. Auto-inducer also for the same species in gram positive, but auto-inducer is cross-species communication, mostly in the bacillus. Okay, so this, yeah. So yeah, this one I was telling you. You have a different range of ASL, like you can have C4 means 4 carbon side chain, or you can have C10 or means longer. This way means you have a lot of variation of different kind of AHL, and they are involved in a different kind of regulation. And yeah, so each bacteria has a different set of these autoinducer molecules. I will not go into that detail, but in general you understand that gram negative has HL kind of system, gram positive have AIP kind of system, which basically looks like this. As I informed you that it will produce this inducer LASI. It is in Pseudomonas aeruginosa. It will keep on producing once they have good number in the outside of cell, they will start binding to LASR, then it will come back and it will activate few genes, it will downregulate some other genes. And this is auto-inducing peptide, where mostly in SRS, here you have propeptide AGRA, and which is secreted outside by this protein AGRB, and it is processed. Here it is not processed, and once it is processed by AGRB, it forms a ring kind of structure on one end. Then it is an active molecule. Here it is not active molecule. After it is processed, then it is active autoinducer molecule. Same way, once it reaches up to some threshold, it start binding with the AGRC, which is having a kinase activity. Kinase activity, you understand? Anybody? Kinase activity? Yes. So it attaches a phosphate to this, then it is a active form of the molecule, and then it goes on, and then one hand it activate this these genes, on the other hand it is activating some other promoter which is normally known as the RNA3, and which is involved basically in the production of different kind of virulence factor and different kind of toxins which is known for SRES, and it is involved in all those. So this is one common signaling which is secondary messenger, cyclic diGMP. So planktonic to biofilm and biofilm to planktonic. There is a, when it is shifting from planktonic to the biofilm, you will have and it is triggered because of some environmental factor or some factors, then you have a higher production of cyclic diGMP. There are certain enzymes involved. I think certainly somebody will be covering this part. You have higher concentration of cyclic diGMP, then they change their structure. Here you can see that they have flagella, right? Here in biofilm they don't have flagella, right? So how they get detached the flagellar structure? Because of cyclic diGMP, it is involved, and it is just one example which is reflected in the phenotype of the bacteria. But there are so many other factors at the genetic level which is changed because of this signaling. And same goes reverse when it is from biofilm to planktonic. There is a decrease in the cyclic diGMP because a certain again EL domain enzymes are involved which cleaves cyclic diGMP, make it two different GTP, and then it becomes planktonic. This is how this circle goes on. So let's just quickly we'll go. I will have a recap whatever I spoke. So you have reversible. You start with the reversible attachment where motility, flagella, and because of mostly chemotaxis, and then you have non-specific physical forces, van der Waals interaction, electrostatic interaction between cell and the surface. Then what is the surface hydrophobicity? What is the charge? What is the roughness? What is the porosity of the surface? All these things significantly decides whether this bacteria will settle here or not. Then once it is reversible, goes to the irreversible attachment, that time it starts changing. They have a growth in form of microcolony. There you have, that time they start producing EPS, and then they have specific interaction sometimes, and ion bridging because of like mostly by the calcium, the hydrogen bonding, quorum sensing plays role. Secondary messenger, cyclic diGMP from here onwards, because once it is activated, then only EPS is produced. And yes, and then maturation of the biofilm, again mostly dominated by the EPS and other matrix component. And then there you have active dispersal. When I say active dispersal, it is not because of some external force. It is bacteria itself which is deciding that okay, now this place is not good enough, we should go somewhere and settle. Okay, in that case they will produce some particular enzymes which will cleave its EPS and degrade EPS thing. That is called seeding. Seeding means it is for from this place to some other place, it is being seeded. So if this place is not good enough, we will go somewhere else and start our colony formation there. Then passive dispersal. Passive dispersal means like sometimes you have a high flow rate of water, or some shear force is involved, or some physical forces are involved that cause slogging or erosion. Changes in the environmental condition, like nutrients are not there, or some high temperature, or many other environmental factors. And yeah. Okay, so this is about biofilm. So since beginning I'm giving the emphasis that we need to understand biofilm. Okay, if we want to get rid away of them, or we want to use them for some benefits, unless we understand the nature of the biofilm, dealing with them positively or negatively is difficult. So definitely we need to study, and how we will study for each stage. You have different techniques, like at the initial stage, adhesion strength, and for the adhesion extent of the bacteria with the surface, you can study different way. AFM is one of them. AFM, which will give you that what is the adhesion force is involved with the bacteria and the surface, and SPR, surface plasmon resonance, that is one of the expensive one, but that gives a very accurate measurement how actively surface is involved in the bacteria adhesion. Then okay, after adhesion and all, you want to see that what is the biofilm mass, and whether how much biomass is viable. You have different kinds of techniques here, like CFU counting, qPCR, flow cytometry, dye staining, okay, XTT, TT, see that I'll cover later. And in total, what is the biofilm biomass? Biofilm biomass means including cell as well as whatever is excreted. Total is called biofilm biomass. There you can use general staining dye CV, which binds with almost everything, and that will give you how much total biofilm there. And then if you become more specific about the to understand what is the biofilm matrix is composed of, protein, polysaccharide, or how bacterial distribution is there. In that case, CLSM, that is confocal laser scanning microscopy, FCS, flow cytometry sorter, that will give what is the distribution of different kinds of bacteria depending on how you label them. And then Excito, there is a different methods for that one. We will not go into that detail. So this is the simplest method of biofilm measurement. So smallest lab can afford that. CV crystal violet is very cheap dye. What you do, you grow in this kind of plate, grow them for some time, treat with CV, wash, and depending on how much biofilm is there, you have a higher or lower CV binding. Okay, that you can measure once you solubilize and you take reading at 570 nanometer. It will give you more or less biofilm formation. Higher the absorbance, simply meaning it is having more biofilm mass. But when I say more biofilm mass, it doesn't mean that more cells are there. Okay, it is in toto means it is including both the things, cell as well as EPS, and that matters sometimes. We'll come to that part as well. We'll see that if you change the ratio of cell to EPS, and whatever property you are measuring will be significantly affected. And but what is the problem with this one is that suppose you grow the biofilm, you have treated with the CV, but this biofilm cannot be used for any other purpose because now it is bound with the CV, and mostly this processing is done outside. And even if you do inside of lab, you cannot use it. Means you cannot do the microscopy, or you cannot further. If you want to grow it further, you cannot. You have to set up a different experiment. So this Calgary biofilm device provides a very simple solution to this problem. Here, if you are able to see some dots, are you able to see from there? Can you see? Here you have some brown color dots, that is small magnetic beads, and here this white thing is cells. So it is mixed thoroughly, and we allow it to grow for some time, whatever our experimental plan is. 6 hour, 12 hour or 24 hour, you want to grow it, and if you want to see whether biofilm is formed or not, because if you see this plate at any moment, it will look fully turbid. It is difficult to know whether biofilm is formed or not, because it is fully turbid, but it is also possible it is still in the planktonic phase, it is not bound. So because growth will be there, what you do, you put a magnet here, below the each well. If biofilm is formed, what happens is this small magnetic beads, they are in the matrix itself, they are stuck, they cannot move anymore. So if biofilm is there, all these magnetic beads cannot come here at the center, and it will not look like this. Okay, if growth is there but it is in the planktonic phase, what will happen, when you put a magnet here, all the magnetic bead will come here and they will look like dot here. When you see a dot, that means it is not forming biofilm, right? Very simple method. And here you are not destroying your biofilm. If you want to grow it further, you give some more time, you don't have to open it, you can continue. And here it is a different kind of system, which is normally known as a Robbins device. So these things, what you do, you pass the media through this one, and these small things here on the surface, you have a biofilm growth. So let's say if you want to monitor whether it is forming biofilm or not at different time point, you can take out one, take reading in whatever you want to take, maybe CV, or you want to stain with acridine orange or any other method, you can do monitoring. And if you are experimenting that way, you can keep on growing them. So you don't have to stall your experiment. And then at some level, with a higher level, if you want to grow, it is known as a biofilm reactor, where these are the facility to give different kind of treatment. Like you can have one control. Suppose I want to see efficacy of any antimicrobial. So one set will be controlled. Here I will add, let's say on different time, if I want to add, or maybe one time I will add, and then I will allow it to grow for some more time, and then we'll see whether it is forming biofilm. And it is simpler version of that same thing, known as a flow cell. Here you have flow chamber. Okay, similarly here also you can have different experimental setup. One can be controlled, one can be treated with the antibiotics, or any kind of treatment, whatever you want to study. And this part you can put under the microscope, confocal or normal fluorescent microscope, and visualize it. And it is advanced form of this flow cell. The flow cell is a big one. You have a chamber of that microscopic slide, so it takes a lot of media as well. But this is just miniature version and advanced level version of the flow cell, which is known as a microfluidics. It is, if you see here, each microfluidic unit is having very similar structure the way you have in the flow cell. Okay, so these are like growth chamber. Here you have inlet for, like one is for bacteria, one is for medium, okay, or some kind of treatment. Then all it goes same way, and here you have outlet. Okay, so each unit is connected here. Like one side you have a medium, and this kind of system is mostly used for longer duration of experiment. When you use 96 well plate, problem is and because most of the bacteria, if particularly pathogenic bacteria, will grow at 37 degrees Celsius. 37 24 hour if you keep, next day what you will see, that 200 microliter whatever you have taken has become 190 or 180 microliter. I mean, there is a significant evaporation. And if you want to extend the experiment for more than 24 hours, it significantly affects your result, because whatever wells you have on the sides, they will evaporate more, whatever you have inside, there you will, it will be less evaporated. The outsider, because there is evaporation, whatever planktonic cells are there, they will also get attached to the wall. And when you take, when you do CV, you will think it is a biofilm. It is not a biofilm. It was planktonic cells which just got stuck because of the evaporation. So that gives a lot of, here you see the side one. It is the same bacteria. It's not that here you have a higher biofilm formation, but only thing it was grown for longer time, 48 hours. So here you have evaporation. Here you can see how thicker CV binding is. So if you take the reading here, you will have a very high reading. You will feel that okay, here you have a higher biofilm growth. It was not higher biofilm because same bacteria, na? Same medium. It will grow same way. But because of evaporation, it will introduce error to your experiment. So these things are taken care because it is completely closed system, and here whatever media you are adding is in millimeters. Sorry, not millimeter, milliliters. Okay. So if you are having 1 liter of this one, it can run for 22 hours and in extremely controlled conditions. Okay, when I say extremely controlled means here you can control temperature, pH as well. Okay, and here you can monitor in the continuous flow system. 96%, once you have to add, it is like a stagnant. Maximum you can do is you can have a shaking. Okay, but here in a flow condition you can have. So yeah, here is just showing that like initially you added inoculation here, then you monitor for some like 60 to 80 hours the biofilm growth, and at the for such kind of system, this thing can be kept under the microscope, whatever fluorescent microscope, if it is having time lapse system or confocal microscope. You set a system because continuously it is flowing. It will take pictures, images, or whatever reading you have set for 72 hours. It is very advanced system of version of the flow cell. Okay, imaging part, when we come, the maximum resolution you can get is in the scanning electron microscope, where you find very detailed structure of this bacteria and biofilm. But one problem is when you prepare sample for the scanning electron microscope, you must have heard you have to dehydrate. You have to dehydrate sample. If water is present, then you cannot do the same analysis. So I mean, again, you are stopping your experiment. If you want to take same image, means you have to stop, dehydrate, then you take same. Though it gives very detailed view of the biofilm, but this is the problem. You are you are destroying your sample. So comes to the confocal laser microscope, which is mostly used for the biofilm studies. Okay, here you don't have to necessarily destroy your sample. Sometimes you can have some GFP expressing bacteria. If it is having GFP expression or some other kind of expression or dye, or even if you are adding from outside, like in the previous system you saw, from one well you can add some dye, it will go and stain the bacteria, and you can have continuous system. So here you are not destroying the sample, and you can have very beautiful and detailed and 3D structure. In SEM, what you see is mostly surface. Okay, surface from where your electrons are getting scattered. You know the principle of electron, right? Electron microscope. Electrons are scattered, and that's how image is taken. You cannot see inside the cell, right? But in the biofilm, in the case of confocal, you can outside as well as inside the biofilm. What is the 3D structure? In the depth, how they are distributed. You see, that's how you can imaging the biofilm. That's how confocal laser scanning microscope is. So why it is possible in the confocal but not in the other kind of microscope is here how we see is here. Let's say here you have a sample. Confocal microscope, it does point by point scanning all over the surface. In the first one, it is called optical sectioning. Then it goes in the second, whatever you set, maybe 1 micron or 2 micron, that you can decide. It will go down, and from there it will do point by point scanning, and at the end you have many confocal slices, and using confocal software, what it does, that it will bring down all the optical slices together, and it will give you a beautiful picture like this. It will have all the 3D information in that one. And how this optical sectioning is done? Basically, when you have any fluorescent microscope, here you see whatever detection system you have. So whatever fluorescent signal is coming from the sample, it is coming from all the depth, okay, and it is coming from all the depth, and all of that is captured in the detection system. And that's why when you see, even though you focus it, but you will find that fluorescent microscopes are, you will see the blur, and that will be almost very thin slice of the sample. But in confocal, what happens is, whatever out of focus signals are coming, it will be cut here. One pinhole is there, the only signal which is coming from a particular point, let's say from this point, that is only allowed to pass, okay, and it goes to detector, and that's where that data is taken. And this is how point by point scanning is done throughout the sample, and at the end all the data is put together, and you have a beautiful 3D structure. Okay, so why one more main benefit of using confocal is, when we are talking about especially polymicrobial biofilm. So polymicrobial biofilm, definition wise, where you have more than one bacteria. In nature mostly it is polymicrobial, as was told in the first lecture. In nature you always find biofilms to be polymicrobial, because more than one bacteria is always involved. Only in lab we try to grow them in a stringent environment when only one bacteria is involved. Other thing we have, because of sterilization is removed. But in nature it is always polymicrobial. And if you want to study that, you don't know how they are distributed with each other. It is also possible that nearby they have colonies, okay, they are not completely mixed, but they are living nearby. You have microcolonies nearby. But the second way can be co-aggregation, okay. In co-aggregation, mostly when they are having cooperation, when they are cooperating, just like suppose you have to live with your roommate, and if he or she is not cooperating, it is very difficult. You will definitely part ways after some time, right? So in this kind of system, once they have cooperation, they can share some resource, or like one bacteria can be degrading one thing which is toxic to the other one. Some kind of cooperation is there. In that case, they have a biofilm like this. We are completely mixed with each other. Then sometimes they have this one, layer by layer distribution. Why? Because some bacteria, they may prefer the solid surface, they may have higher affinity for the surface. Okay, so what they will do? They will form a monolayer. Then the second or third bacteria finds it very easier to settle on a one microbial colony rather than the surface itself. Okay, in that case they have distribution like this. Okay, so first one will quickly summarize that as I told that they are ubiquitous, they are almost everywhere, and they have a very complex structure, and EPS matrix is mostly composed of polysaccharide, eDNA. They have regulation, definitely, particularly quorum sensing, and I think it is one of the listed topics. You will have more detailed quorum sensing lectures, and yeah, once you know how this biofilm is working, you can have targeted disruption and few advanced imaging things that we discussed. So this is the future of biofilm, because last almost 20-25 years people are working on the biofilm. Quite understanding has been developed, but it is time now to, as per the understanding, we should be able to engineer them for our use. That I will be covering in the second part. And multi-omics integration means you combine all the data, whatever you get from the genomics, proteomics, and put together and understand. Okay, so this is end of the first one. Anybody wants to ask something?
Anyone having questions?
Shall we proceed for the second one, or will already 3:10? We'll continue and finish that. If there is no question, I will move to the second part. Or anybody wants to take break? 5 minutes. We'll move on. We'll start second one. Okay, we'll reassemble after 5 minutes then. Okay, we'll start the second part. Few things will be similar to that, so I will not go into that detail because it is back to back. And so implication of biofilms. So topic is microbial biofilm, food processing, health and disease. So in all these three areas, you will see negative impact of the biofilms in all these three. But there are some good biofilms as well. That I will talk. Okay. So they can be good when you are using their robust nature for your some applications. Bad in some case, like mostly in disease and all, and sometimes ugly, especially where you have very persistent infection. So and they are it is affecting without us realizing that how much it is affecting our various part of life. And how it is affecting, if you look at the how much billions of money is involved in biofilm management and all. In food processing, 324 billion, right? Healthcare, 387 billion. Wastewater treatment, which is now coming up as a technology to treat wastewater, 117 billion market. Agriculture, here one more is not mentioned, is the biocorrosion, where biofilms are involved in the corrosion. And corrosion means almost all the industries are getting affected because of corrosion, and large extent it is because of the microbial influence part. So it is extremely important to deal with biofilms, understanding how much significantly actually they are affecting our life. As I said, they have some particular nature, particular characters, and they are you can use them as a two sides of the coin. So bad means like biofilms, biofouling in tertiary system. This kind of picture Professor Arun sir was also showing you. In our power plants, we face this kind of problem where initiated by the biofilm, then it goes up to the biofouling. You can see that it is almost clogging the system. This is heat exchanger where heat is exchanged, and that's how electricity is generated. It is completely almost 40%, 50% is blocked. Brings down the plant efficacy, bring down the electricity generation efficacy, and then you have a microbial induced corrosion. So because every year so much corrosion is happening, and when it is happening, industry have to have replace those parts, and it costs millions of dollars or rupees, you say. But because they have certain characters, they can be used as a good biofilm, especially in catalyst in many industries. And many biofilm based bioremediation, heavy metals, polycyclic aromatic hydrocarbons. If you will see in example wise that it is much better system as compared to the planktonic if you want to use them for this purpose. Then you have ugly clinical biofilm, especially diabetic food infection. Similarly, as you see biofilm grows, you have a contamination, then colonization, then there is a stage when it is almost impossible to get rid of a diabetic food infection. That becomes so much prominent. Ultimately, amputation is the only option left. So that kind of problem these biofilms create. And it is also on the medical devices, catheters, and that's where persistence creates a lot of problem. So this I have already talked recently, so I will not go into that. So let's talk something good about biofilms. Okay, otherwise we will think that they are only villain. They can be friend as well. They can be very good when it comes to bioremediation. Why? Because it is very robust system, so they can act as a living catalyst for many kind of organic synthesis, and can be used for clinical applications. And it is natural, not by us, but it is natural, very good, beautiful nexus of ecology and engineering, where they can act as a living biocatalyst for performing challenging conversions for many industries. So first of all, what is bioremediation? Definition wise, you use any bioprocess. Okay, it can be enzyme, or it can be a living organism, planktonic cell, or any biological system to mitigate toxic pollutants. Okay, that is called bioremediation. Whole cells are effective. I would not say that they are not effective, but downstream processes are difficult, and that adds additional cost. Whole cell means, let's suppose that you have a bacteria which is able to degrade some particular thing, or it can able to, or some in some case pigments are having very commercial use. It can produce. In that case, it can be useful. But like after once it is produced, you need to remove those whole cells or bacterial cells from the whole system. Then only you will have purified product. So purification is a problem there. So that is the additional cost. So to that, up to some extent, people use immobilization, because we have to anyway remove it. Let us immobilize the whole cell. So it will retain the cell. We can reuse again for the next batch process. But again, it has, when we use immobilization technique, we use many chemicals. It will affect up to some extent its viability, or even bring down the enzymatic activity. Or people have gone, okay, whole cell immobilization is a problem. Let us isolate the enzyme, and that enzyme itself, because that is the thing which is doing the job, will immobilize the only enzyme. Yes, it works. It can be again reused for the next batch, but again, downstream, because when you are using some process for the industries, you always look at the cost. Effectiveness is one thing, but cost is always, which is the one of the major thing for taking up that technology. So chemical binding you can do for the enzymes. Whole cell you can either entrap or encapsulate in like calcium alginate. So the question comes, for all these things, naturally immobilized microbial, whether they can be a solution, because they have all these required solutions for the for any process. How biocatalysts are different from the enzymes or whole cell? They have a advantage that they, when they are in the cells, they are more stable, and in the cell you have a different expensive cofactor. Otherwise, you have to add separately. And enzyme purification, you don't have to do it here. It is already present in the cell. So all those things brings down the cost. Okay. Whole cell process is mostly confined to batch or fed batch process, and due to substrate or whole product or toxicity, short-term biocatalyst stability, and complexity of the running large-scale chemistry. What it means is that when you are using a whole cell, whatever you are giving as a substrate or the product which is being formed, that can have a toxic effect on the cell. Okay. So best thing is that if you are having a continuous reactor operation. Okay, and yeah, so reduce those complexity, you have this immobilization away, what I just told you. So but still it has some limitation, like when you are immobilizing enzyme or any whole cell, it will definitely reduce the absolute activity of enzyme or the cell. And then again, there is a problem of diffusion mass transfer. Okay, limitation of substrate and oxygen loading reaction rate, because all these process definitely brings down up to some extent. And then there is a lack of universally applicable immobilization concept. So one method cannot be applicable to all the process. So if we look at the biofilms, it is protected, as I told you, that they are less susceptible as compared to the planktonic or whole cell. So you have, means up to some extent it is already protected, and then increased availability nutrients for growth, because EPS is there. Even you are having some toxic treatment, still a lot of nutrient is available by which it can repair itself, it can grow itself, and then in some cases you need to have good amount of water. There it plays a role. And here the best thing for any industrial process, that you need not to separate it from the bulk liquid waste. You have system on biofilm system, whatever treatment you are giving, getting treated, that it is getting separated. So, but despite all these features and almost 2 decades of the biofilm research, still if you look at very few biofilm processes are in the industrial scale. So what we need to do is that we can just look at what are the recent developments and how we can use it for the biorimidation, and we need to understand the characterization technique, because as I told that for each biofilm you have to understand how it is being regulated. Okay, what kind of treatment will affect in what way. If you don't understand, optimization is very difficult. And yeah, so fundamental research and industrial application are two different things. Okay, you have to see what is needed and what is needed for the industrial application. Both the things you have to see, and then you have to come to some common point where both the things can be trade off. Okay, so if you look at the literature, you will find that nowadays people have used heavily for biorimidation of many toxic pollutants, halogenated organic compound, pesticide, petroleum products, azurides. Many of this one is already done at our lab itself, and heavy metals, okay, and polyaromatic hydrocarbons. If you look at particularly poly pH, in short we say aromatic, right, that benzene ring, okay, very difficult to degrade by any bacteria. And when it is polyaromatic, means 2 or 3 benzene rings are fused together, phenanthrene, pyrene, that becomes for a normal bacteria it's very difficult to degrade. But in the when they are in the form of biofilm, it helps. I'll come to that how it helps. Biofilms are much more efficient when it is coming to the degradation of pH. So they have particular these characters, like they produce EPS, and there is a if you want to regulate sometimes like we want to increase the EPS production, there we know how quorum sensing is affecting the EPS. We know whether we have to, how much we have to increase quorum sensing. By from outside we can give quorum sensing molecule and increase its structure. Then comes some population of dormant state cell and VNPC state cell. It is not good thing. A good thing for one thing, that it is okay resistance to the it is not getting affected because of the toxicity of the product or the substrate. But problem is you have biomass. If you have 10% of the total biomass which is not metabolically active, sometimes it was benefit, but here 10% biomass is a waste, right? Because it is not contributing to your industrial process, whatever you want to do. And VBNC state cells, it is that not VBNC that you read. Here it is that biomass which is not actively involved in the bioprocess. So these two things, anyway, it is good for the biofilms, but when it comes to the industrial applications, we have to take care of these two as well. So EPS, like extensive biomass, as in other thing we see that heat transfer or mass transfer, that also we have to see. It should not become too thick that our product itself doesn't go inside, our substrate it doesn't go inside and gets converted. Okay, and VBNC state cells means these are the unproductive biomass and low specific category. So these are the negative where biofilms are having negative impact. These two things are beneficial to us, but the same two things, when we want to use them for a benefit, these two factors play a negative role. So if you want to use biofilm as a biofilm catalyst too, these are the two things what we need to look. First of all, they should able to form good biofilm at given substratum of interest with sufficient amount of EPS, allowing for high biofilm robustness on one hand, but low mass transfer limitation on the other hand. So these two characters we have to come to a point where both things can be managed properly. And biofilm grown cells should be genetically stable. Genetically stable in particular in those cases where we are modifying bacteria, where we are like you have a cloned product, okay, where you have inserted some particular gene for some degradation. So it should not be like that, okay, today we have grown the biofilm, but that whatever bacteria is having, over the generation it is getting diluted. So whatever plasmid you are using, or it is getting integrated into the genome, it should be stable. Then only you can use it for the industrial applications. And it should be highly porous. Highly porous means here it is opposite, because you are trying to use them for some purpose. So your substrate should be able to reach to the cell so that it can be converted. So, yeah. So again, this characterization comes into the picture, that you have to understand how spatial distribution is there, distribution of the metabolically active cells, and what is the EPS matrix is composed of, whether it is protein or exopolysaccharide, what is the thickness, what is the topography. All those things have to be optimized for a process, and and then response to the relevant stress. Sometimes like shear forces. Sometimes we have seen in lab that if you give little bit of shear force, your biofilm grows much better in that shear force, because it senses that okay, some kind of stress is there. I should produce more robust biofilm, so it will start more EPS. So for characterization, there are different techniques. So for biofilm component, there are different methods. Like ATP method, it will tell you what is the total biomass and actively divided cells, not the VBNC. Biofilm thickness, electrochemical method, that where you use the impedance, because biofilm, as I told you, that it is 90% water, which is not a very good conductor of electricity. So thicker the biofilm, you have more impedance, and that's how you can measure the biofilm thickness. If you remember in the microfluidic, I was showing one picture for the impedance. So just by using impedance, you can know that how thick your biofilm without treating with any stain, without doing any kind of measurement. Just impedance will tell you how thick your biofilm is. And for characterization Raman microscopy, it will tell what kind of functional groups are involved. And yeah, so definitely all the techniques have some advantage, some disadvantage, depending on your requirement and all those things. You have to choose what you want to do. And these are the microscopic technique, AFM, SEM, ESM, where you don't have to completely dehydrate your sample as in SEM, and it will give you good image. CSLM, as I told you, and CSLM can be used in a fantastic way, okay, depending on what is your purpose. And you can use different kind of fluorescent dyes. Like if you tag with the cytonine, it will tell you about what is the cell vitality. It's very simple, fast, and fluorescent reporter. Sometimes you want to monitor growth over the period of time. Tag some housekeeping gene with the fluorescent reporter, and it more the biofilm, more the you have a fluorescent, and then that's how you can see. And this kind of thing is mostly in the genetically engineered organism. Natural organism means you cannot do that, obviously. Reporter gene means you are putting it in some vector or something. Okay, so fish can be used especially for the if you are looking for the what is the microbial diversity in your microbial biofilm. And yes, it can also detect VBNC state, because VBNC, as I told you, that if you grow them in lab, it will not see anything on the plate, only few bacteria will grow. But if you are using fish or even 16S sequencing, you can know that 100% of bacteria, what kind of bacteria are involved. This is very what do you say, complicated and good technique for some parameters if you want, particularly because it is completely different food processing. Now I have not shown that we have set up multiple reactors across the country, especially for STP. Somebody was asking whether we can use for STP. So biofilms can be grown in the form of granules. Granules are just like a ball, small, this much ball, and that is very, very effective in water treatment. And there you see, because it is granule, it is a just EPS and cells. It is not attached to anything. Okay, there is no solid surface required. What we do, we provide a CS force continuously for few days, and all the biofilm will become a form of granule that can be used for the STP or any kind of treatment. So if you see, like it is in millimeters actually. I wish I would have put one picture of that. So if you see from the surface to, if you go toward the core, on the surface you have the different parameters. If you see the oxygen concentration, it will be high on the surface, but as you go deep down, you have anoxic environment inside at the core, and this is just showing the surface of the granule. So and that is why granules are being used for almost any kind of treatment. There you have thousands of bacteria, different kinds of bacteria, and almost till date we have whatever we have given for the treatment, it shows that it is able to degrade. Why? Because it has such enormous diversity. If it requires aerobic treatment, surface cells will take care, because here you have higher oxygen concentration. If it needs anoxic environment, the whatever cells present at the core, they will do the anoxic reaction. In one system you have both kind of bacteria, and it can take most of the degradation part, because sometimes it needs oxygen, sometimes it doesn't need. So if you see across the surface, parameters of oxygen, pH and all those things changes, that you can monitor, and it is required to know that, especially for academic purpose, if you want to link the way whether oxygen or anaerobic environment is involved in some particular degradation, we need to know that how oxygen or any other parameter is changing across the surface. So what we do, this is it is a micro needle. You have a needle of 10 micron. You cannot even see by naked eyes how thin it is. You insert inside and connect with the reference electrode. It will give you measurement in the volt that you can convert into the this concentration of your whatever you are looking for. Very good technique, but problem is being very fragile in micro, and this is made of glass. Okay, and by mistake if you hit the surface, broken, it will go. 1 lakh rupees is gone. Each needle cost almost 1 lakh rupees. So very good and sensitive technique, but yes, you need to be expert to use that. So we have like this oxygen, pH, sulfide gas, thermo, and one reference electrode. So this is overall for industrial purpose. If you want to study in a different method, like basic understanding using flow cell, and as I told you, some rotating disc bioreactor or different kind of systems, you see how biofilms are performing. Some observation you have to do, visualization using mostly confocal microscope or microfluidics, and then you have you want to see that how topography is changing, because topography is very, very important, especially when it comes to the treatment, any kind of treatment. So topography means that up and downs of the surface. Okay, that you have very flat surface, obviously less surface is exposed to the your aqueous solution or treat whatever you are treating. You have more rough surface, means more surface is exposed. So in that case you have always better change. So here you see, I'll just come to with quickly some examples why if you understand biofilms, how it helps. So this is one example where we degraded phenanthrene using pseudomonas biofilm. In the presence of calcium, it shows that you have more biofilm. Okay, but as I told you, more biofilm means it can be more EPS or it can be more cells, both. If you do CV, you will not know, but if you do confocal, where you can here you can see that it is showing that it is more EPS production. Okay, and here what you are seeing is this side EPS to cell ratio. Okay, same thing is same bacteria, but depending on EPS to cell ratio, it is having a different capacity to degrade phenanthrene. Okay, so sometimes if you increase the biofilm formation, not necessarily that you will have a better degradation or product formation. For this thing, because you need active degradation, you need more cells, right? So in that case, when you have a lower EPS to cell ratio, that environment is more favorable for the phenanthrene degradation. Okay, So this is just one example where you see pyrene degradation. Here you see biofilm 1 is able to degrade much better as compared to the planktonic. One example of heavy metal removal that we do in our lab, basically we deal with the uranium a lot, and we are using biofilm for uranium removal. So if we see that one method can be that uranium 6, which is soluble and more toxic, can be converted to uranium 4. But the problem is it is easily reoxidized. Just now we were discussing that heavy metal is not that easy. Reduction is the best way possible because that brings down the toxicity, but it is not that easy because most of the time we have aerobic environment, and in such cases again it comes back to the original state, then again it becomes soluble. So uranium Deinococcus biofilm we have used, and this one you see it is present in the presence of calcium. As I told, calcium most of the time it increases the EPS production, and here uranium it is being absorbed because of the EPS. Okay, so when you have a calcium in the biofilm growth, you have more EPS production, more removal of uranium. Okay, we will skip to that. Okay, so this is some characterization that we wanted to understand, that why it is forming good biofilm. Otherwise, the wild type Deinococcus does not form biofilm, and that is why even though we were doing I think some 20 years research on Deinococcus in BART, but coming to the industrial level it was difficult because same problem. It is a cell, it is not forming biofilm. So you had to isolate the cells after any kind of treatment. But if you have biofilm formation, that problem is resolved. So that is happening with this bacteria that we have modified in the lab, and it is GFP marked, so you don't have to scan it. Whenever you see under microscope, you will see a green fluorescent this one, and it forms very good biofilm as you can see. So that this one we have used for the what previous slide you saw, that it is taking up uranium beautifully. So this strain is being used. Wild type is not able to take up. So just we want to understand what is happening actually, why it is forming biofilm. So as we treated biofilm proteinase K, DNase, alginate layers. Proteinase K will cleave the proteins, surface proteins. DNase will cleave all eDNA if it is participating. Alginate layers will cleave mostly the exopolysaccharide. So if these things are present in the biofilm, I mean protein, exopolysaccharide, respective enzyme will be effective, right? So in the case of proteinase K, it is dispersing the biofilm. Simple meaning is some kind of protein is involved, right? Simple. So mostly it has a SLPA in the surface. We suspect that this might be involved, and some we did some more detailed work. I will not go into detail, but this was the protein which is found to be responsible for the this one, I mean biofilm formation. And if you increase the DNA concentration, SLP formation also increase. So that's why it was showing much better removal in the presence of this one. So okay, so again same problem, that it is, even though it started forming biofilm in the presence of calcium, it is more stabilized, but it is not good enough. As we see that I was discussing that granule, it is still not able to form granule. So some solution is that we can immobilize and we can use it for the treatment. So it is able to remove, but again the advantage you get in natural biofilm is that you can reuse for 10 times, 50 times, 100 times continuously. When you immobilize, their restriction happens. Up to 3 cycles it is doing fine, but after 4th or 5th it is not possible. There is one reason that whatever bioabsorption sites are there, that will be saturated because of uranium binding. If you have continuous growing biofilm, then you don't get saturation, because again and again some new cells are coming which will have some new absorption site. But when you try to immobilize, you face this problem. In different system where polyacrylamide gel is used, there we have very good removal, but it has a different practical problem. It will get clogged after 3 or 4 treatments. So solution is currently we have switched to that will try to mix many bacteria with known properties. Okay, so because we have a number of bacteria, we have seen that okay, this particular bacteria shows very good EPS formation. This one is having good radio tolerance. I mean Deinococcus, and someone is having uranium removal. All those bacteria, if we put together and make a system, okay, where we know that which bacteria are involved, that kind of system may be useful. So that's what we are calling synthetic polymicrobial biofilm. So natural biofilm means it will have a lot of bacteria, maybe hundreds or thousands of bacteria, where known or unknown both kind of bacteria are involved. Here we are having synthetic, where we are trying to add and try to manipulate which are known. This I told that this kind of different kind of systems, but again when we are moving to the polymicrobial system, further we have to understand a few things before we proceed, like what kind of interactions they are having, what kind of spatial distribution is there, whether there is a nutritional cooperation or competition, right? So although sometimes one bacteria inhibit the growth of other bacteria. If suppose I am thinking that okay, these 4 bacteria I am trying to put together, but one bacteria may be inhibiting the growth of other bacteria. Okay, so all those things we have to first cross check, confirm that okay, it is working, then only we can move further. So you see, with these 4 bacteria we are using, most of them are all 3 are tagged with some kind of fluorescent protein, so that we can monitor them under microscope whenever is required without giving any dye treatment. They have up to some extent some competition is there, or one bacteria is overgrowing, other bacteria, especially Deinococcus, is slow growing bacteria. It reaches to log phase after 48 hours. But most of the bacteria, if you inoculate 6, 7, 8 hours, you will reach the log phase if you have grown them anytime. So these kind of things are there, where you have to optimize how much cells, in what ratio they should be mixed, so that they don't outcompete the other one. They should coexist with each other. And this is the one example. Like you can see that some combinations, they are showing better growth, other are showing lesser growth, and compared to individual biofilms. But good news is that even after all this, because we know that they are very good at uranium removal, in most of the combinations they are able to achieve almost close to 100% removal. So, and these are the metagenomic study that after 15 days or 1 month, how their composition is changing. So we have done good understanding this. So this is able to remove because 98% is removing. We have now in the process of developing this polymicrobial biofilters. So here we are trying to put all these known bacteria. Both the side you have activated charcoal, which will take up many organic content before and after the biofilm treatment here, and then it can be taken here, because we are thinking of a very applicable system, where you have a simple system, like you just put the water from the top, and from the down you have a purified one, especially low concentration uranium contamination is there, like in Hisar groundwater is reported that low level of uranium is there. So for that kind of water, simple, you just pour the water from the top, from the down, because most of the uranium will be taken up. You can use that, and here you have filter now, so you don't get this bacterial contamination in the water. So this kind of system we have almost developed. Shall I cover this one? I'll try to finish in 5 minutes. So one aspect I think nobody else will talk about is biocorrosion, because mostly it will be on health or on infection and food processing. I'll just very quickly give what is biocorrosion. Corrosion, right? Corrosion, So basically, if you understand that microbial induced corrosion means you have natural process of corrosion, okay, and presence of bacteria can just influence. Term itself it tells that it can influence the rate of the corrosion. Okay, if you have any chemical reaction, that side you have product, and if that product is removed, the this whole equilibrium will be shifted toward the right side. This you must have read in the chemistry, right? The same thing happens here. Most of the product, it is taken up by the bacteria, and that's how this biocorrosion is enhanced. So this is normally what happens. So in biocorrosion, three things are involved. One is material, which is being degraded, and microorganism, of course, and the aqueous medium. Okay, and medium means what is the pH, oxygen, redox condition. All these things affect biocorrosion. So when it comes to biocorrosion, one of the most prominent bacteria is known as the sulfate reducing bacteria. So here you have a biofilm where see this iron is taken up by the biofilm. It is deposited as the APS. That's how the increase in the corrosion process happens. So this is just electrochemistry of the how this corrosion is being influenced. So they look corrosion, normal corrosion and biocorrosion are very similar, but there are few difference, basic difference. Both the case you need oxygen and pH is playing a role. So in normal corrosion, which is a natural process, here biofilm formation happens when we talk about biofilm corrosion, and it is metabolically involved, because most of the time when biocorrosion is happening, it's not that only bacteria is present is called biocorrosion, but bacteria is mostly involved in the corrosion process because it is either taking up the product or it is increasing the acidity. Simple thing you can maybe knowing that when bacteria grows, sometimes you will see the drop in the pH because of organic acid production in the system. So you know that if it is acidic, corrosion will enhance because of higher H plus concentration. So if you look at the corrosion in detail, at initial stage it may be different, at later stage it may be different. Both the things are biocorrosion, but if you look at the mechanism, it is little bit less different. So at like initial stage where you don't have a full biofilm, where just some bacteria are getting attached to the surface, most of the surface is exposed. It is stainless steel surface which has a passive film of chromium oxide. Okay, so it may be degraded up to some, and when such kind of system is there, here cathodic reaction is because oxygen is present on the surface. Okay, so here cathodic reaction will be oxygen plus water, and in the presence of electron it will form 4OH, and because chloride is anyway it is, I don't know, you know that there are different kinds of corrosion: pitting corrosion, crevice corrosion. Chloride is mostly involved in the pitting corrosion. Pitting means sharp pit is formed. Crevice means it is like general one, on the surface it goes. Crevice is really, pit is like very deep. So there chloride is involved, and this kind of corrosion happens when you have a initial stage of the biofilm where your surface is exposed, or once you have full biofilm formation. Okay, so as I was telling in the case of granule, once you have thick biofilm, outside it may be oxygen rich, but inside it will be negative, or you can say anoxic region where oxygen is not present. So there it cannot happen. This cathodic reaction will be replaced by different bacteria, a different reaction where like sulfur reducing bacteria, they can perform this reaction. They can take hydrogen and they can make hydrogen. One very good example recently that we did is Bacillus subtilis biofilm, which was shown to cause biocorrosion in borated 3,4 stainless steel. borated is used in our nuclear setup, where we keep the fuel rod, and fuel rod means it doesn't produce electricity, means that much is not there, but it is still very radioactive. It is kept in a water pool, and from there, I mean, because radioactivity cannot come out, but that structure is made of stainless steel. So recently people have shifted to borated. Why to borated? Boron, you must have heard very recently during this nuclear India, I mean India-Pak recent war. Have you heard anything about boron? No. Some messages were there that plane is coming with the boron. Have you heard? Why boron? Because boron acts as a moderator in the when radioactivity is happening. So that is why it is recommended that if you use the boron stainless steel, it is borated. It can absorb radio activity very efficiently. But the problem with the borated stainless steel, it is more prone to the corrosion. And then we don't know how, if some bacterial contamination there, how it will affect. So this one is, I'll just see this graph. I will not go into the detail. You have a borated stainless steel control, and with the this bacteria, this one is showing the corrosion rate. So you can see without bacteria, this much is corrosion is happening. In the presence of bacteria, it is showing this much. Okay, so many people say that bacteria, people are unnecessarily talking that bacteria is involved in corrosion and all. When they don't find any reason, they simply blame bacteria. No, it plays very important role. There you can see. And this is the mechanism. I will leave it. I will not go into that detail. But why this biofilm helps, because biofilms help in creating this kind of gradient of oxygen. Okay, outside you have a oxygen rich zone. Here you have a hypoxic zone. So when you have hypoxic zone is there, then you have a separation of cathodic reaction and anodic reaction. That is how pit formation is increasing, and it can affect this one. Okay. So quickly I'll summarize this one, that as I told, that it can play positive and negative role depending on what we are looking for, and it has a lot of bioremediation potential. Clinical challenges are there. It can have industrial impact, corrosion particularly, and biofouling pose significant economic cost, and that's what we have moved to engineered biofilms. So now we are engineering biofilm to achieve what we want. Yeah, thanks. So sorry, I had to rush. Yeah, I understand that many things, because I have rushed, you may not have followed, but feel free to contact me. I think on the first slide I have given my contact email. If needed, you can take from Professor Gunjan. You can contact me for any biofilm related work in future as well. And if some of you want to do any project, we take July to December 8 students, and from January to June 8 students. So obviously are not from the same center, but we have projects for this one. So if you are interested in any biofilm related work, you are free to contact me. Any quick questions if anybody wants to ask? Yeah.
Is it possible to design a universal anti-biofilm agent that disturbs only the pathogenic activity and leaves the beneficial ones?
Repeat your question.
Is it possible?
Universal? It should be universal, but it should kill the pathogenic, but leave
biofilms without harming the beneficial ones.
Very difficult. Very difficult. So if you have a, that's what you always have to trade off between specificity and efficacy. Like you want to use, you want to kill everything, you take nanoparticles or chlorine. That's why it is being used, still used in the almost all the plants, because it is non-specific. It will kill everything whatever is there. And still most of the cooling towers, wherever biofilm problems are there, people are using chlorine. Two reasons: it is non-specific, quite effective and cheap. So whenever it is coming to industry, economy is the major thing which plays role. We have a better actually oxidant like chlorine dioxide. That is more better than chlorine. In fact, chlorine is having problem of generation of it generates THMs, trihalomethanes, which is carcinogenic. Still people are using because it's very cheap. Chlorine dioxide is 5 times more effective than the chlorine, but still because economy, they have industry have to see which is more and effective as well.
Thank you, sir.
Okay, no more questions? Okay, no problem. You can afterwards also you can write if you have any confusion. If you don't ask here, don't want to look stupid that what is the foolish question. Okay, because many people don't ask in public because they feel that I should not look stupid. Okay, so you can write to me. I'll respond to you. Okay. Yeah.
Okay, so on behalf of Central University of Haryana department, I thank Dr. Sudhir Shukla. So we have been in touch for last 10-15 days maybe only. So and he accepted our invitation to come here. He has to rush back because his flight is at 10. So maybe we can contact him later if you have some queries. So thank you, Dr. Sudhir. Thank you for this visit. Okay.